Hello everyone, welcome to Physics Wallah English YouTube channel. I'm Shalini Somashekar, your botany teacher. I welcome you all to today's lecture. Let me know in the comments how are you all doing. Hi Abhishek, Anjali, Om Singh, NEET Crazy Girl. Good morning to everyone. How are you all doing? How's the preparation coming along? Hi Dopamine, so nice to see all the old students back again. Good morning. How's everyone? What's the status of your preparation? Hi Anjali, good morning. Hi Rishikesh, good morning. Hi Priya, Uma, Tara. Good morning everyone. So I'll briefly tell you what the agenda of this series is. You all know we've already finished something called the 50-day NEET challenge, where we went in depth with each chapter, we discussed concepts and we also solved relevant questions. And in this series, which is called the NEET Most Important PYQ series, we are going to be solving previous year questions that we feel are very important, and we are going to do this chapter wise. So we will be solving around 500 questions per subject, as I had announced in the short video, and it will be done in two parts. So today's is the first part of botany, which I will be handling. In today's lecture, we are going to be covering all of your grade 11 chapters, alright, and in the second part, Chaitra ma'am will be covering all your 12th grade questions, PYQs. I hope everyone's ready. Thanks for asking, Dopamine. I'm doing great too. Yes, Rohit, it's been so long and I missed you all so much. Alright, everyone's ready. Let's get started then. Okay. So everybody, I'm sure, would know by now all of the chapters that are included in your 11th grade syllabus, but let me just go through it once. Your first chapter is Living World, and then you have Biological Classification and Plant Kingdom, Morphology in Flowering Plants, Anatomy of Flowering Plants. Then you have Cell, the unit of life, Cell Cycle and Cell Division, Photosynthesis in higher plants, Respiration in plants, and Plant Growth and Development. These are the chapters from which we will be solving PYQs today. So before we start attempting the questions, I request you all to please enroll yourselves in the Unity portal. The link must be on top of this chat box. It must be pinned on top of the chat box. So please click that link and enroll there so that you can participate without any hassle on the poll, right? Alright, so Priya is ready. Everyone's ready. Let's get going. Please remember to enroll in the Unity portal. Alright, so before we start off, let me just tell you about the number of questions we've been seeing since the last few years from each of these chapters. From the Living World, we don't see a lot of questions appear. It's already a very small chapter, very basic, very a chapter that you can easily understand. So in the past few years, we have seen around one question, sometimes no question has been asked from this chapter also. And from Biological Classification, you can expect around 2 to 3 questions. From Plant Kingdom, Morphology, Anatomy, Cell, the unit of life, you can expect around 3 to 4 questions. And from Cell Cycle and Cell Division, this is the chapter that carries the highest weightage in your 11th grade syllabus. This chapter is very small, very easy to understand, a lot of things to memorize and remember, exactly what happens in which phase, all of that you'll have to remember. But yeah, once you remember all of that, once you memorize all of that, this is easy peasy lemon squeezy. So all of those 5 questions you can easily answer and get your full marks. Then you have Photosynthesis, Respiration and Plant Growth and Development in your last unit, which is your Plant Physiology unit. From these chapters, you can expect around 2 to 3 questions, right? NEET aspirant is asking what Unity portal is. Hi sweetie, hi Sebastian. Unity portal is basically where you'll have to register in order to participate in the poll. As we are conducting, as we are discussing questions, MCQs, I'm going to display each question and give you like 30 seconds to 45 seconds to answer those. And you will be typing the correct option in the chat box. Depending upon how quickly you type, depending upon how many questions you answer correctly, we will get a leaderboard at the end of the session. So if you want to participate in that, you'll first have to enroll in Unity. Otherwise, your answers will not be accepted by the poll. Alright. So students who've been attending classes since the beginning will know about this. For any people who is this is the first time you're joining, please enroll in Unity. Yes, Rohit. Good morning, Ansh. Good morning, Mayuri. All right, let's get started with our first chapter without any further delay. The first chapter is the living world. It's a very simple, very easy chapter. So in your NTA syllabus, NEET syllabus that the NTA has released, this is all you'll have to know for in the chapter, the living world. It includes what is living, biodiversity, what is the need for classification, what does taxonomy and systematics mean, what is the concept of species, taxonomical hierarchy, binomial nomenclature, and all of that. In the previous textbook, the one that was not rationalized before, you had another concept called taxonomic aids that has been deleted from the NEET syllabus this year. So this is all you'll have to know. Let's get started with the questions. Wait, before that, I'll tell you how many questions have been asked from this chapter over the years. As you can see, there are a couple of years where even one question was not asked from this chapter. Other times you can see one question. In 2016, phase 2, 3 questions were asked from this chapter alone, which is, that hasn't happened again. So that is the weightage of this chapter, the living world. So I'm going to start the poll right now. Let's get started with the questions. Is everyone ready? Let me know. Give me a thumbs up in the chat box. Let me know. Is everyone ready? One simple instruction before we start solving the questions. I want you all to attempt each question as if it is a question in your final NEET exam. You will read it very carefully. You will understand the question very carefully and answer in the chat box. Okay. I'm not ready to listen to any of your excuses saying I did not read the question properly. That is why I did not answer it correctly. We have very few days left. Usually when I'm conducting classes, when I discuss questions, I read out the question myself. So what happens then is I kind of emphasize on what are the important terms. But today I'm not going to do that as well. I'm going to start the poll, and reading the question is also your job. I'm not going to read it for you. How quickly you read it, how correctly you read it, all of that will matter. All of that you should practice before your final NEET exam. So let's get started. I think everyone's ready. NEET aspirant is asking, how do I poll answer? Just look at the question, understand it properly. Whatever you think is the correct answer, just type that option in the comment section. You don't have to write the yeah. You don't have to write the entire statement. You just have to type whether it is A, B, C or D. What you think is the correct answer, just type it in the chat box. If you've enrolled in Unity, that will get registered in the poll. Alright, let's get started. And here is our first question, which is a pretty simple question. You shouldn't be taking more than 20 seconds to answer this, but let's see how many of you all will get it correctly. I'm not going to read the question. It's your job. You have to do it yourself. This is a question that was asked in NEET 2019. As I already mentioned in the beginning of the session, we are going to solve past year questions that are very important. Alright, most of you are saying C. So C itself is the correct answer. 68% of you all have answered correctly. Ansh, Ram and Shubha are in the top 3 spots. So this is basically a question based on binomial nomenclature. Everybody knows that Carolus Linnaeus or Carl Linnaeus, who we call the father of taxonomy, he is the person to have first introduced binomial nomenclature, right? So in binomial nomenclature, we basically give names to living organisms scientifically, where each organism's name will have 2 words. The first word is called the the generic name, and the second word is called the specific epithet. There are a couple of rules that govern naming of organisms. What are they? First of all, the name has to be derived from Latin. If you can't derive it from Latin, we are going to Latinize it. And then whenever you're printing it, it has to be italicized. Whenever you're writing it with your hand, whenever it's handwritten, you have to underline it separately. And then the generic name will always start with a capital letter, and the specific epithet will always start with a lowercase, right? And whoever has first described that plant, the author, their name can also be used as a third word in an abbreviated form. So Mangifera indica is our mango tree or the mango plant that was first described by Carl Linnaeus. So Lin is the abbreviation for his name. So we can write it as Mangifera indica Lin. So the correct answer here is option C. I don't think there's any doubt here. All right. Okay. Now let's move on to our next question. This again is based on the concept of binomial nomenclature. So read carefully. I've already explained the rules in the previous question itself. Read carefully and answer correctly. I'm so tempted to read the question because I'm so used to it, but you should get the practice of reading it properly. So I'll refrain myself, hold back and try not to read the question. Okay, most of you are saying D. I saw that someone had also said A. So what happens in such questions is, okay, let's read the question first. This is from NEET 2019 that was conducted in Orissa. Which of the following is against the rules of ICBN? Here we have to find out which is that rule which is not according to the rules of binomial nomenclature. In the first option, we have handwritten scientific names should be underlined. Of course, whenever we are writing it with hand, we have to underline it separately. That is true. Every species should have a generic name and specific epithet. Yes, there will be 2 words in each name, which is why we call it binomial nomenclature. This is also true. Scientific names are in Latin and it has to be italicized whenever it's printed. That is also true. Generic name and specific names should be written starting with small letters. Specific epithet, of course, starts with small letters, but the generic name always starts with a uppercase. It starts with a capital letter. So this statement is what is incorrect. So the correct answer to this question is option D. All right, 81% of you all have answered correctly. Shubha, Ram, Rishikesh are in the top 3 spots. Now let's move on to our next question, which is again based on the same concept, so you can understand how important this concept is. Every year, pretty much the same questions have, similar questions have been asked. Yeah, Rohit, I'm trying to hold back so hard, like I'm not used to staying quiet. I can't shut my mouth, that's why. Nomenclature is governed by certain universal rules. Which one of the following is contrary to the rules of nomenclature? Contrary means not according to, opposite to the rules, against the rules of nomenclature. Generally, from binomial nomenclature, these are the kind of questions you can expect. Option A, the names are written in Latin and are italicized. When written by hand, the names are to be underlined. Biological names can be written in any language. Right now, if you look at option 1 and option C, they are contradictory, right? We are saying in one place it has to be in Latin, in the other option, in the other statement, we are saying that it can be in any language. So, 2 contradictory statements, both of them cannot be correct. One of them have to be incorrect, right? What is option D? The first word in a biological name represents the genus name and the second is a specific epithet. That is true. What is not true here is that you cannot call it, write it in any language. It has to be derived from Latin or Latinized. So the correct answer here is option C. Hopefully 100% of you, no, not yet. Hi Suresh, good morning, long time, how are you? Suresh, Nikhil and Ishan are in the top 3 spots. Yeah, Rohit is a very regular student. Here is our next question based on taxonomical hierarchy. There is something wrong in this question. I want any of you to identify and point it out after reading this question. Something about this does not seem right. Whatever it is, you have to find out and let me know. Hi Anurag, hi Sundara Pandyan and Vishal. I'm good too, Suresh. Okay, so we have finished this question. Let's discuss it. Who found out what is the problem with this question? Has anybody noticed what the issue with this question is? Is there an issue at all? Okay. In the taxonomic categories, which hierarchical arrangement in ascending order is correct in case of animals? Yes, Shubha is right. Actually, in all of the options, it's starting with kingdom, so this is descending order. If you look at the different categories in the taxonomic hierarchy, we have kingdom, phylum, class, order, family, genus, species. When we dealt with this chapter in your 50-day NEET challenge, I had given you this mnemonic, which I'm sure every one of you will know. Keep plates clean, or family gets sick. This is one easy way for you to remember all of these in the correct order. Right? Over here, kingdom is your highest category and species is your lowest category. Everybody knows ascending and descending order from math, right? Ascending is when you're arranging something from the smallest to the largest. Descending is when you're arranging something in the reverse order, largest to the smallest. In the question, they've asked about ascending order, but in the options, everything is descending. So that is the problem, but apparently no grace marks was given for this question. Yeah. So what is the correct answer? Keep pots clean or family gets sick. Option A is the correct answer. I hope everyone has answered correctly. All right, brilliant. 100% of you have answered correctly. Only 14 students are participating or what? I'm seeing around 42 questions live right now. Where are the others? Why are you not taking part? We have Ram and Vishal Prithviraj and Rohit Gupta in the top 3 spots. Congratulations, guys. Okay, there is some cricket fan who has a cricket mnemonic for the same thing. Whatever works for you guys, whatever helps you remember better, remember that. Okay. All right, let's move on to our next question. Which one? Oh, wait, I'm not supposed to read the question. I'm sorry. Everyone says A, and it is A indeed. So where did this come from? You know, in your textbooks, in your NCERT textbooks, you have this one table. In this table, you have the classification or the categories of 4 different organisms, 2 animals, 2 plants. 2 animals, one is man and one is housefly, and the other one is mango and wheat. They are for plants. So it's important for you to remember all of that here. At the first glance, it may seem like it's a little difficult, but we all know that man is a chordate, housefly is arthropod. In animal kingdom, you've studied pretty much all of these things. And Mangifera indica, everybody knows. Triticum aestivum, you know. So it's not very difficult. You know that wheat is a monocot, mango is a dicot. There are just a few things from this that you'll have to memorize. Once you do that, any question from this table, you'll be able to answer very easily. Congratulations, everyone has answered correctly. Praveena, Dopamine and Ram are in the top 3 spots. Today I'm noticing how Ram is answering most of the questions correctly and also is in the top 3 spot. Congratulations, Ram. Suraj is typing something in Hindi which I don't understand. Okay. I don't understand Hindi, Suraj. Okay. I tried to read, but I didn't understand. So sorry. I don't know if it's anything important. You can expect questions like this also, which was asked in NEET 2016 in the second phase. Okay, if he's motivating us, then thank you, Suraj. Oh my god, for this question I'm getting a mixture of A, B, C, D. Phylum is Arthropoda. Family. Option A is the correct answer. Okay, 65%. Vishal, Abhishek and Pavan are in the top 3 spots. This is where it becomes important for you to read this properly, memorize this properly. Okay, so whenever questions like that are asked, you can understand well. I mean, you can answer correctly. Here is another question that was asked in the year 2016, Phase 2 NEET. NEET aspirant is asking how you can check your rank in the leaderboard. After each chapter, I will go through the entire leaderboard so you can see where you stand. For now, I'm just going through the first 3 names because it will consume a lot of time if we have to go through it all after each question. After each chapter, we'll go through the entire leaderboard. Okay? Okay, most of you are answering it as C. Also, don't worry about how quickly you have to answer. Make sure that the answer is correct. Okay? Answering late but correctly is much better than answering quickly and getting it wrong. Right? There are 4 statements given to us here. Definition of biological species was given by Ernst Mayr. That is correct. Photoperiod does not affect reproduction in plants. That is incorrect. Actually, this is from a chapter called Plant Growth and Development. In Plant Growth and Development, you had a concept called photoperiodism and vernalization, dormancy and all of that, but in your rationalized syllabus it's deleted, and also in your NTA's NEET syllabus it is deleted. So photoperiod basically is, of course you would have learnt in your 11th grade last year, right? Because last year it was not yet rationalized. So photoperiod is basically the leaves of plants kind of measure the amount of daylight they receive and how much darkness they receive. Depending upon that, they will initiate flowering. So that is photoperiodism. It does affect reproduction in plants because it influences production of flowers. Flowers are the reproductive organs. So of course, photoperiod in many plants will influence flowering and reproduction. So that is incorrect. Binomial nomenclature system was given by R.H. Whittaker. No, not at all. It was proposed by Carolus Linnaeus. R.H. Whittaker is responsible, or he is his contribution is basically to in the 5 kingdom classification. He proposed the 5 kingdom classification. So that is incorrect. Binomial, okay, that's incorrect. In unicellular organisms, reproduction is synonymous with growth. That is correct. Because every time they divide, reproduction is happening, growth is also happening. So that is correct. In the given statements, A and D are correct. So correct answer here is option C. Let's see how many of you got it correctly. 91%, not bad. Anurag calling himself Bhai, Anurag Bhai and Pavan and Suresh are in the top 3 spots. Now let's take a look at our next question. Suraj, I'll share the notes as soon as this class is over. In about half an hour or 1 hour, you can expect the notes. You'll get it in the description box of the same video. Like, yeah. Rohit is asking how long it's going to be. It's probably going to go on for at least 5 hours, in my opinion. I hope everyone's had breakfast and come. The common characteristics between tomato and potato will be maximum at the level of their family, order, division, genus. Actually, the correct answer is option D. Not many of you have answered. Okay, so here's the thing. This was asked in 2013. This was asked in 2013. Tomato, potato, both of them have the same generic name. Over here we have to understand one concept. As and when you go from the species level to the higher levels, to the kingdom level, the number of similar characters is going to decrease. So the similarity in characters is highest at the species level, and as you go up the taxonomic categories, up the levels, it's going to go on decreasing. Okay. So here, okay. Both of these have the same generic name. Tomato is Solanum lycopersicum, I think, and potato is Solanum tuberosum. Both of them have the same genus. So maximum level characteristics will be common in the genus level itself. Alright, so correct answer here is genus. Regardless of which two are there, remember that as and when you go up the taxonomic hierarchy, up the levels, the similar characters is going to decrease. For example, if you consider within the same species, humans, we all are the same species, we have very high degree of similarity, right? We all have 2 arms, 2 legs, 2 eyes. All of these are very similar. The bone structure is very similar. The physiology of our different organ systems is very similar. But if you compare our species with another species, for example, if you compare us with a chimpanzee, though we might be closely related, but there will be more differences. If you compare humans with dogs, there is going to be difference. So within the same species, similarity is going to be highest. As you go up, it's going to decrease. All right. Rohit has a question. He has a doubt. Going from kingdom to species, the same character increase, so why not genius is the answer. I didn't understand your question. Could you please rephrase it? Shani, Sunny, whatever your name is, I don't know. Apologies if I've pronounced it incorrectly. In NEET, you do not have math, right? This is exclusively for NEET. So you'll have physics, chemistry, botany and zoology. Each subject in 2 parts. Today's first part of botany. Likewise, you'll have first part of zoology, chemistry, physics, and then second part of each of those subjects. Math will not be included in this series. I hope that's clear. Okay. So here, correct answer. Okay, so I want you to understand this one concept that is given to you in your NCERT textbooks. It is given in a very dispersed manner. I have picked it up from different places and made a table out of it. I want you to memorize whatever you see in this table and in this table as well. If at all there are questions asked from these 2 concepts, you should be able to understand it very easily. Mahesh, thank you. Okay, so there are, when you study about the taxonomic categories, you have 1 paragraph for each of those categories, right? So you have species, and under that you have a couple of examples, and then you have genera or genus, and under that you have a couple of examples. So it's kind of scattered there. I put it all in the form of a table, different table for plants and different table for animals. In your textbook, they've mentioned Solanum tuberosum, Solanum nigrum and Solanum melongena. Solanum tuberosum is your potato. Solanum nigrum is something to do with darkness, black, all of that. So there is a plant called the black nightshade, that is your Solanum nigrum, and Solanum melongena is your brinjal. Brinjal is also called eggplant, it's also called aubergine in some countries. So all of those are the same name. All of their scientific names, biological names are given here. I want you to notice how all of them have a common genus, which indicates that they are closely related, right? So all of these belong to a family called Solanaceae. Okay, this Solanaceae along with another family called Convolvulaceae belong to the same order, polynomials. This is given to you in your NCERT Okay, so remember this, memorize this. Where is your video for glycolysis? Sagar, it is, what do we say? Work in progress. My job is done. The editors are doing their job. As soon as it's done, we will be releasing it. Okay, hi NEET aspirant. What is your real name? Rohit asked, I'm sorry, I don't think I saw your question. Oh, Cartoon Clips is also back. Welcome back. Okay, so this you should memorize. You will have this in your notes. Please go through it and remember. Okay, next let's take a look at all of the animals and the different things mentioned in your NCERT textbook. You have lion, tiger and leopard mentioned. Lion's scientific name is Panthera leo, tigers is Panthera tigris and leopard is Panthera pardus. Notice how all of them have the same genus. They are very closely related. You look at it and you know they all are big cats. And so they will have the common genus. Cat, they have mentioned the scientific name of cat is Felis in your textbook. Felis domestica is not given here. Domestica I have added. Okay, so that is your domestic cat. This Felis along with Panthera belong to the same family called Felidae. Dogs belong to a family called Canidae. Felidae and Canidae together, they belong to the same order called Carnivora. Primata is another order in the animal kingdom which includes primates like your monkeys and gibbons and gorillas, right? So this Primata and Carnivora orders belong to the same class Mammalia. Similar to Mammalia, you have many other classes in animals, which is your birds or aves and reptiles and amphibians and fishes or pisces. All of these belong to the same phylum, which is Chordata, and Chordata is one phylum in the kingdom Animalia. Okay, so wherever it's highlighted yellow color, those details of those organisms you don't have to worry about. This is all that is mentioned in NCERT, so go through this table, memorize everything there is over here. So if at all any questions are asked based on this, you should be able to answer it without much difficulty. NEET aspirant's name is Yogesh Kumar. Okay. I think I teach like myself, not anybody else. But anyway. Okay, so that's it. We're done with the chapter, The Living World. Let's go through the leaderboard once. Ansh is on top of the list. He's answered 6 out of 8 questions correctly. And then we have Suresh and Ram. All of them have answered 6 questions. The difference in this leaderboard is based on how long they have taken to answer the questions, their average time duration. But for now, I don't want you to worry about how much time you're taking. In your NEET exam, it does not matter how quickly you answer. All that matters is whether your answer is correct or not. So don't worry about this. Don't try to compete and fight against time. Try to read correctly, answer correctly. Okay, so that is what you should be doing. Congratulations to everyone, all the 3 of you. And we have Abhishek and Rohit and Shubha, Vishal, Mayuri, Ishan, Pavan in the top 10 spots. And we have Dharmesh and Yogesh and Priya, Sweetie. Okay, I think they answered a couple of questions and left midway. Okay, so this is pretty much your leaderboard. Let's move on and talk about the next chapter, which is... Hi Tejaswini, good morning. No problem, better late than never. Okay, Sweetie has come in between. Got it. I hope so, Rohit. Let's see. All right. So 5 kingdom classification. In biological classification, we are going to study about 5 kingdom classification. Salient features of 3 of those kingdoms, Monera, Protista and fungi. And then we are going to learn about lichens, viruses, viroids, prions and all of that. All right. Now, let's take a look at the trend. Previous years, how many questions have appeared from this chapter? In 2023, unfortunately, there were no questions, but in the last few years, you can see one question 22, 21 and 2020. Before that, more questions used to be asked, 3 questions, sometimes even 5 questions. But of late, the number of questions that are appearing from this chapter has reduced. Okay, so that does not mean this year there will be no question from here. It does not mean that you can take this chapter lightly and not, revise, because this year the number of chapters itself is less. Many chapters are deleted, many concepts are deleted. There are chances that from chapters like living world and biological classification also more questions can be asked. Okay. Yes, cartoon clips is absolutely right. Please do not socialize in the chat box. Okay. All right, NEET aspirant. Bye bye. In today's class, Naina, we are going to finish all the first grade botany chapters. 10 chapters we are finishing today. Ansh has a question. If we go from kingdom to species, the specific characters increase, right? Okay, so if you didn't understand, let me give you an example using this same table. Okay, so we have kingdom Animalia. Ansh, this is for you. We have kingdom Animalia. In kingdom Animalia, you have a mosquito. In kingdom Animalia, you have a sponge. In kingdom Animalia, you also have a human. Among these 3 groups, is there more similarity or less similarity? The group is huge. The number of organisms, the number of species in that group is huge. So, between 2 individuals, similar characters might be less. If you consider the same for species, if you're considering one single species of mosquitoes, one mosquito will have more resemblance with another mosquito. One human will resemble more with another human. One cat will resemble more with another cat. So when you look at the species level, there's going to be more similarity. As you come down from the kingdom to species, the number of similar characters is obviously going to increase, right? Have you understood? Ansh, have you understood? I hope you have. So let's start off with the questions from biological classification. Ready everyone? Give me a thumbs up and let me know. Yes, everyone's ready? Okay. Here's our first question. For those of you who have joined late, please enroll in Unity. Otherwise, your answers will not be registered in the poll. Hi Chandan, good morning. I'm doing great. Thank you so much for asking. NEET aspirant, I thought you were going to vote. This is a question that was asked in 2022, NEET 2022. The first statement is, mycoplasma can pass through less than 1 micron filter size. So when we're talking about filter size, we generally talk about the pore size. So in a filter, you will have pores, right? For example, the filter that you use to strain your coffee or tea, that will have pores, right? So in these filters which we use to filter microorganisms, the pore size is going to be like super tiny, very small. It can measure in microns. So in the first statement, they have said that if there is a filter and the filter has a pore size of about 1 micron, then will mycoplasma pass through it? Actually, this statement is not directly given in NCERT. In NCERT, that they have mentioned that mycoplasmas are the smallest cells, but their size, the dimensions is not mentioned. So this is here they have told that they can pass through less than 1 micron filter size. They can. Usually their size will be less than 1 micron, so they will be able to pass, but this dimension is not given to you in your NCERT textbook. This statement is a correct statement. Statement 2, mycoplasma are bacteria with cell wall. No, mycoplasma are not bacteria with cell wall. They lack a cell wall. So this statement is incorrect. Okay, so among the given 2 statements, statement 1 is correct, but statement 2 is incorrect. So the correct answer here is option C. Hopefully everyone's answered correctly. Alright, not bad. 81% have answered correctly. We had NEET aspirants, Sweetie and Ram in the top 3 spots. PMS. The PMS I know is... Never mind. Alright, so what are halophiles? Halo is salty, phile, philia is liking for something. Halophiles are those bacteria that are seen in areas that are extremely salty, even saltier than oceans and seas. So this is option 4. Thermoacidophiles are bacteria that live in extremely acidic and hot areas like your hot water springs. And then methanogens live in the guts of ruminants. They are involved in the formation of biogas. Cyanobacteria are, they are basically blue-green algae that require some amount of moisture. They live in aquatic environment. Correct answer is A4, B1, C3, D2, which is option A. Okay, 89% have answered correctly. Rohit, Suresh and Harshit are in the top 3 spots. Please to all the NCRT diagram in one shot, please. Okay, I will discuss with the team. If there is a feasibility, we will do that as well. Now let's take a look at our next question. Very simple questions from this chapter. Which of the following are found in extreme saline conditions? We just discussed that. Eubacteria, cyanobacteria, mycobacteria, archaebacteria. Okay, D is the correct answer. Archaic, archaea is basically ancient. These are organisms that evolved on Earth at a time when the conditions were very extreme, not like it is today. So they are archaebacteria. In the top of the leaderboard, we have Harshit and Yogesh and Sagar in the top 3 spots. So correct answer here is archaebacteria. If you have to be specific, then it is halophiles. Halophiles are a type of archaebacteria. Okay, so correct answer is option D. Let's take a look at our next question here. Absolutely. Cartoon clips is right. Which among the following are the smallest living cells known without a definite cell wall, pathogenic to plants as well as animals, and can survive without oxygen? Yeah. It is mycoplasma. Correct answer is option B. 97% correct. Ansh, Sweetie and Sagar in the top 3 spots. Let's move on and take a look at our next question. Very simple questions. This was asked in 2017. See, very similar questions. This just shows how frequently similar kind of questions are asked. Lokesh, unfortunately, the poll will not pick up emojis. You have to type in the correct option. Correct answer is archaebacteria. I've already explained it. Still, some of you have answered it incorrectly. Sweetie, Varshika, only studies, what a beautiful name to have, are in the top 3 spots. Next question. It shouldn't take you like more than 10 seconds to answer this. This was asked in the year 2015. Cell wall is absent in nostoc, aspergillus, funaria. Nostoc. What is nostoc? Nostoc is a blue-green alga. In nostoc, what is the cell wall made up of? It is Monera. It belongs to Monera. So the cell wall is present and it is peptidoglycan. Made up of peptidoglycan. Aspergillus is a fungus and the cell wall here is made up of chitin. Funaria is a moss. The cell wall would be made up of cellulose. So it is mycoplasma which lacks the cell wall. Correct answer is option A. I expected 100% to answer correctly, but I don't know what is wrong. 93% of you all have answered correctly. We have Sweetie, Dopamine and Ansh in the top 3 spots. Congratulations, guys. Yes, I think I'd already mentioned in the 50-day NEET challenge, Rohit, if you remember, but I've added those slides here as well. For algae, when we come to algae, I'll tell you what the trick is. All right. Let's move on and take a look at this question. I think we'll get 100% correct answer for this question. Maybe not. Sai Krishna, you don't have to type your name every time. You can just type A, B or C. Very similar questions, Harshit. This is the reason we should look at PYQs and analyze. You know how all of these, oh my god, what have I done? Okay, 95% have answered correctly. Sweetie, Pradeepa and Ansh are in the top 3 spots. So you may have seen how whenever these cricketers, I'm sure most of you follow cricket, that is why I'm giving you this example. Whenever they are playing cricket, they don't just look at the strengths of people in their team, right? Of course, you'd know who is your best bowler and all of that, who is the best batter. You would know all of that. But it's also important to know about the opposition's strengths and weakness. Whichever team you're facing, you should also know and learn about those players' strengths and weaknesses so that you can plan your strategy according to the strength of that team as well, which is exactly what we need to do in competitive exams. You should look at the paper previous years, how the questions have been asked. Is there any pattern that we see? Are some concepts more important than the other concepts? Here, it's like you're playing against the paper. So unless you know and you've known and understood your paper very well, you will not be able to face it properly, right? So that is where it's very important for you to analyze your past year questions. I don't know what Sagar is talking about. Samdani is asking me how I'm doing. Thank you so much. I'm doing great. All right. Here is our next question. Hi Lisa. Welcome back. It's been long. Yeah. Also, whenever you come across questions like this in your exam, identify which is the correct statement, find out which is the incorrect statement. Sometimes our mind, because it's in a hurry, it's anxious, it's nervous, you might read words incorrectly. Your brain might read the words incorrectly. So whenever you come across questions like this, please underline the term right there so it kind of gets fixed in your brain and you are going to look at the correct statement, find the correct statement and not the incorrect one. Which of the following is a correct statement? Option A, cyanobacteria are a group of autotrophic organisms classified under the kingdom Monera. That is correct. Bacteria are exclusively heterotrophic organisms. Right now we can see how statement A and statement B are contradictory. Cyanobacteria are autotrophic, we are saying, and in the next statement we are saying bacteria are exclusively heterotrophic. If bacteria are exclusively heterotrophic, we can't have autotrophs in that group, right? In bacteria, in Monera, you can see a very diverse mode of nutrition. you have autotrophs, you have heterotrophs as well. So they are not exclusively heterotrophic. Exclusively heterotrophic means only heterotrophic, right? So this is not true. Slime molds are saprophytic organisms classified under kingdom Monera. No, slime molds are unicellular and they are eukaryotic. They are classified under the kingdom Protista. Okay, so that makes this incorrect. Mycoplasma have DNA, ribosome and cell wall. They have DNA, they sure will have ribosomes, but they lack a cell wall. We've discussed this same point, I think, in at least 3 questions before. So this also is incorrect. The correct answer over here is option A. Varshika, Ram and Sagar are in the top 3 spots. Okay, thank you, NEET aspirant. Harshit is asking me to provide tough questions. Tough questions we have already discussed in the 50-day NEET challenge. Like I told in the beginning of the session, the idea or the agenda of this whole series is to help you understand what kind of questions appear from what chapters in the past years, to make you more aware of the kind of questions asked in the actual NEET exam. More difficult questions, tricky questions, we've solved more than enough in the 50-day NEET challenge. So you can go there and watch those lectures. And I've also given a lot of homework questions in those sessions. If you download those notes, you'll get a lot of those questions which you can solve. Okay. So today's agenda is not to solve difficult questions, but today's agenda is for solving PYQs. Okay. Pani Puri Golgappa. Okay, fine. Samdani, I think I'm pronouncing your name correctly. Whether it will be tough or easy depends on you. It depends on how well you are prepared. So if you're prepared, anything will be easy. If you're not prepared, it will be difficult. I would say it will be relatively easy because you have fewer chapters to study. Many chapters have been deleted even in physics, chemistry and biology as well. Since you have relatively a lesser load of chapters, you'll have to learn fewer concepts. That will make it easier for you, right? But it depends on how well you've understood the concepts and all of that. Till what days this series would continue? Lisa, I'm not very sure about the other lectures. Mine will happen today. Other botany class, Chaitra ma'am will handle. Zoology, Vindhya ma'am and Chavi ma'am will handle. About physics and chemistry, I'm not really sure. I don't even know the schedule. I only checked my class. You can check the community post or the community section on this YouTube channel. You'll get it. You'll get the schedule over there. Okay? All right. So let's move on and take a look at our next question. I see a lot of B's, a couple of A's. Lokesh gets very excited whenever it's option B, so he can use that emoji. Okay. Which of the following organisms are known as chief producers in the oceans? What are producers? We've learned in ecosystem chapter. Organisms that can photosynthesize, prepare their own food, they are the producers. But if you look at it, dinoflagellates, diatoms, cyanobacteria, sometimes euglenoids, they all are capable of performing photosynthesis. They all are producers. But the question is, in the ocean, which are the chief producers? This is a statement directly taken from your NCERT. So it is diatoms that are the chief producers in the oceans. Correct answer to this question is option B. Only 76% of you all have answered correctly. Vishal, Tejaswini and Varshika are in the top 3 spots. Congratulations, guys. Let's take a look at our next question now. This is from 2018. You may have, I'm sure you'd know by now that NEET as the single all India entrance exam for medical colleges, for medical courses, is happening from 2016, right? Initially from 2016, 17, 18 and even 19, a lot of questions used to be asked from outside NCERT syllabus. Some questions, some concepts that are not mentioned in NCERT, even those from those concepts questions used to be asked. But over the years, you have seen how that has reduced drastically. Of late, most of the questions are asked from the NCERT textbooks itself. So this is one of those questions that was asked outside of NCERT. So ciliates are basically a type of protozoans which use cilia for locomotion, right? So how are these ciliates different from the other protozoans? Which are the other protozoans? The other protozoans are your flagellated protozoans, amoeboid protozoans, and then you have sporozoans. They are your other types of protozoans. How are these guys different from those is the question. In the first option, it's given they use flagella for locomotion. Ciliates do not use flagella for locomotion. It's very evident from the name itself. They use cilia for locomotion. So this statement is incorrect. Having a contractile vacuole for removing excess water, this only ciliates don't possess. Even other protozoans which don't have a wall of sorts, which live in aquatic environments, they will also have contractile vacuole. So this is incorrect. Using pseudopodia for capturing prey. Pseudopodia is not used to capture prey by ciliates. Pseudopodia, amoeboid protozoans will have. So that is incorrect. Having two types of nuclei. See, I mentioned that this is a question which has some concept that is not included in NCERT. That is this point. Ciliates actually have two types of nuclei. One small nucleus called micronucleus and a larger nucleus called macronucleus. So this is one of those few questions which used to be asked outside your NCERT syllabus. Okay, so the correct answer here is option D. Nobody has answered correctly, but it's all right because it was not part of your syllabus. I completely understand. It's all right. Ansh, what is this? Is it clear now? It's okay, Jyoti. All right. Here is our next question. Please read the statements correctly. Here you have to identify the statement that is wrong. All of the statements are regarding diatoms. Are the walls of diatoms easily destructible? No, they are not. Why are they not easily destructible? Because they are embedded with silica, right? So first option itself is incorrect. Option B, diatomaceous earth is formed by the walls of diatoms. That is correct. Diatoms are the chief producers in the oceans. We solved a similar question based on this statement itself previously, I think 2 or 3 questions ago. So this is also correct. Diatoms are microscopic and float passively in water. They do not have any locomotory structures. Along with the water currents, passively they float. So correct answer here is option A. This is the incorrect statement. Option A is the correct answer. We have Sweetie, Lisa and Sanya in the top 3 spots. All 3 girls, congratulations. All right. Here is our next question. Chrysophytes, euglenoids, dinoflagellates and slime molds are included in the kingdom. There's confusion between C and D, I think. Most of you are answering it as D, but some of you are answering it as C. Correct answer is option D. Just 76% of you all have answered correctly. Vishal, Sagar and Lisa are in the top 3 spots. Chrysophytes, euglenoids, dinoflagellates, slime molds. Who can tell me what's common to all of those? They are all unicellular, they are all eukaryotic. All unicellular eukaryotic organisms are included under kingdom Protista, right? So when you consider all living organisms, everything that is prokaryotic is included under Monera. All of the unicellular eukaryotes are included under Protista. All of the multicellular eukaryotic autotrophic organisms are grouped under Plantae. Absorptive heterotrophy, fungi. Ingestive heterotrophy, animals. So yeah, remember. Right, Sagar. No problem, Lokesh. As long as you know that you're answering correctly, your concepts are clear, it's fine. It does not matter. Here is our next question. Read carefully. The statements are a little lengthy. Jayesh is asking to teach in Hindi. Unfortunately, my Hindi is very poor. You cannot understand my Hindi. And also, this is a pure English channel. The name of the channel itself is Physics Wallah English. I'm sure there are a lot of students here from South India who cannot understand Hindi. So, in this channel, everything will be taught in pure English. I'm sorry, I cannot help you in that regard. Okay, I see B, C, most C's and 1 B. Let's see. We have to find out which is correct. Okay. Some of the organisms can fix nitrogen in specialized cells called sheath cells. Are they called sheath cells really? Which are those organisms that can fix atmospheric nitrogen? They are our cyanobacteria or blue-green algae, right? What are those cells called? We call them heterocysts. If you remember, there is a diagram of Nostoc given in your NCERT textbook where one of the cells is also labeled heterocyst. You thought it's asking for the incorrect one. Harshit, that is what I've been saying since the beginning of the lecture. Please read the question carefully. Every time such a question is answered, in your mind, first look and confirm whether they're asking for which is the correct statement or the incorrect statement. It's very silly. Such mistakes you can't be doing. Okay? Next, fusion of two cells is called karyogamy. Fusion of nuclei is karyogamy. Karyon means nucleus, its fusion is karyogamy, so this is incorrect. Fusion of protoplasm between two motile or non-motile gametes is called plasmogamy. Yeah, this is correct. Organisms that depend on living plants are called saprophytes. No, saprophytes depend on dead and decaying plant and animal matter. So this also is incorrect. We were asked which is the correct option. So the correct answer is option C. 70% of you all have answered correctly. Anshika and Mayuri and Tanmay, again all 3 girls in the top 3 spots. Congratulations. Is there any doubt in this question? That's what I've been telling you. You have to read all the questions as if it is your final NEET exam. Do not take it lightly. Okay? Samdani, I'm from Karnataka. Here is our next question. I'm not going to read it. It's a statement based question again. I don't want you to tell me you thought it was correct, you thought it was incorrect. Please spend time in reading the question correctly. That is the most important thing that you have to learn to do. Reading quickly and reading correctly without making any mistakes. Okay, I see a lot of A's and a couple of B's and a C. Which of the following statements is incorrect? Yeasts are unicellular. This is incorrect. Morels and truffles are edible delicacies. Yes, they are. Claviceps purpureus is a source of many alkaloids like LSD. You may have learnt about this in Human Health and Disease as well. Conidia are produced exogenously and ascospores endogenously. Option A is correct. This is also correct. Conidia are exogenous, ascospores are within the asci, that is endogenous. Correct answer for this question is option A. 81% have answered correctly. We have PMS, Harshit and Neha in the top 3 spots. Let's move on and take a look at our next question, which is a match the following type of question. This is a question from 2019. What happened, Lisa? What are saprophytes? Very simple, like, oh, very easy questions. So far you have seen how the difficulty level of questions in both these chapters is not very high. They are pretty much easy, pretty much direct questions. We have prepared you for even tougher questions in the 50-day NEET challenge. So it should not be, difficult for you to approach your final NEET exam. What are saprophytes? They are involved in the decomposition of dead organic materials. A is 1. Parasites live on living plants or animals. 3. Lichens, symbiotic association of algae and fungi. Mycorrhizae. Myco means fungi, rhiza is about root. So they are symbiotic associations of fungi with roots of higher plants. So this is 1. Wait. Saprophyte, symbiotic association. This is 2, right? A is 2, B is 3, C is 4, D is 1. A 2, B 3, C 4, D 1. Correct answer is option A. 70% almost have answered correctly. Varshika, Ram and Tanmayi are in the top 3 spots. Here is our next question. Which among the following is not a prokaryote? You know, sometimes when students by mistake, instead of typing ma'am on the chat box, they write mama. I find it very endearing. I used to do that a lot when I was a student myself. I used to call out amma in class instead of ma'am. Which of the following is not a prokaryote? Yeah, option A, that is eukaryotic, unicellular. Option A is the correct answer. 93% have answered correctly. Lisa, Sagar and PMS are in the top 3 spots. I have close to 300 slides. I do not know when we will finish. We are in the 38th slide yet. After karyogamy followed by meiosis, spores are produced exogenously in sexual spores produced exogenously in basidiomycetes. Which of these is an example for basidiomycetes? Mushroom agaricus. Option C is the correct answer. 86% of you have answered correctly. Varshika, Soni and Anshika in the top 3 spots. Soni is so confident, he or she is already calling themselves a doctor. Congratulations. Option C is the correct answer. So here I have added a slide where you have examples of so you're going to have this in your slide. Use this to memorize all the examples. Here is our next question. All right. So we have to find out which one is incorrect. Are all fungi eukaryotic? Yes, they are. Do they possess a purely cellulosic cell wall? No, they will have a cell wall made of chitin. They are heterotrophic for sure. They are both unicellular and multicellular. Unicellular example, as Sagar is saying from the beginning, is yeast, and multicellular, the remaining fungi. So here the wrong option is option B. 83% of you have answered correctly. We have Aanchal, Jyoti and Anjali in the top 3 spots. Here is our next question. Yes, it is a polysaccharide. I think the spammer is taken care of. I wonder what they get. Which one of the following statements is wrong? Eubacteria is also called false bacteria. No, it's called true bacteria. Phycomycetes are also called algal fungi, correct? Phyco itself relates to algae. So, algal fungi. Cyanobacteria are also called blue-green algae, correct? Golden algae are also called desmids. Desmids are also called golden algae or golden brown algae. So, here the incorrect statement is option A. Huh? Why can't I see any answers? What is the glitch? Let's look at our next question now. One of the major components of cell wall of most fungi is cellulose, hemicellulose, chitin and peptidoglycan. Cellulose and hemicellulose you mostly find in plant cells. Chitin is what you find in fungi. And peptidoglycan in bacteria. So correct answer here is option C. 86% have answered correctly. Sanya, Aanchal and only studies are in the top 3 spots. Next question. The imperfect fungi which, okay wait, I'm not supposed to read the question. The imperfect fungi which are decomposers of litter and help in mineral cycling belong to Phyco, Asco, Deuterobasidiomycetes. Which of these? It is Deuteromycetes. They are also called fungi imperfecti. Because we don't know how they reproduce sexually, we only know about their asexual reproduction so far. That's all that has been discovered. So 76% have answered correctly. Aanchal, Lisa and Sanya, top 3 girls. Congratulations. This was a question asked in 2015. Yeah, Ansh. Lokesh is very fond of using emojis. But unfortunately, it will not be picked up by the poll. So you won't know where you stand. See, like I mentioned, Samdani, we are taking PYQs. These are not questions that we have created. The questions that we have created and discussed, all the difficult, tough type of questions is already done in the 50-day NEET challenge. The idea of this PYQ series is to make you familiar with the kind of questions that have been asked in the last few years of NEET. Okay, so I hope you understand. Rohit, there are a little over 300 slides. Here is our next question. Select the wrong statement. Cell wall is present in members of fungi and plantae, right? In fungi, you have a cell wall made up of chitin, and in plants, you have a cell wall that is usually made up of cellulose, right? Mushrooms belong to basidiomycetes. Yes, they do. Pseudopodia are locomotory and feeding structures in sporozoans. No, sporozoans lack locomotory structures. Pseudopodia is present in amoeboid protozoans. So this is the incorrect statement. Mitochondria are the powerhouse of the cell in all kingdoms except Monera. Yes, this is also true because in Monera there are no membrane bound organelles like mitochondria. So correct answer to this question is option C. What? What went wrong here? What did you think is the correct answer? What do you think is the correct answer? A and D. Why? What is Janta Palte mean? Did you understand this now? Why have so many of you made mistakes? Only 2 people have correctly answered, Abhi and only studies. Next question. Which of the following is correct about viroids? Do they have RNA with a protein coat? No. Do they have free RNA without protein coat? Yeah. They have DNA with protein coat? No DNA. Correct answer is option B. Okay, 78% have answered correctly. Sweetie, Lisa, Ram in the top 3 spots. Let's take a look at our next question from 2019 Orissa NEET. Mad cow disease. Mad cow disease in cattle is caused by an organism which has it's also called bovine spongiform encephalopathy. Mad cow disease. It is caused by prions. What are prions? They are proteins that are abnormally folded and infectious. So correct answer is option B. Whenever students have doubts in such questions, what they do is, okay, they will look at which 2 options are similar. So they will think 2 options are very similar, probably one of them is correct and take either C or D and then they'll get -1. So correct answer here is 91%. Soni, Lisa and Vishal are in the top 3 spots. Let's move on and take a look at our next question, which is also based on prions, I suppose. No, it has prions, viroids, everything. It's a statement-based question. Prions are abnormally folded proteins, yeah. Viroids lack a protein coat, yeah. Viruses are obligate parasites, correct. Infectious constituent in the viruses is not the protein coat, it's the genetic material which could either be RNA or DNA. So correct answer here is option D. What? Only 38% have answered correctly. Lisa, Sweetie and Anjali. Why? I see most of you have answered it as D, but somehow it's not reflecting in the poll. There must be some glitch. Hi Geetesh, good afternoon. No wait, it's still morning. Good morning. Here is our next question. Viroids differ from viruses in having. What do viroids have that viruses don't have is the question. DNA molecules without protein coat, RNA molecules with protein coat, RNA molecules without protein coat, DNA molecules with protein coat. Correct answer is option C. 96% have answered correctly. Thongni, Lisa and Aegis, I think I pronounced your names correctly, are in the top 3 spots. Here is our next question. Yes, and that RNA is of low molecular weight as Geetesh points out. Which of the following statements is wrong for viroids? Do they cause infections? Is their RNA of high molecular weight? Do they lack a protein coat? Are they smaller than viruses? Yes. Potato spindle tuber disease, all of those is caused by viroids only, no? So they cause infections. Their RNA is not of high molecular weight, it is of low molecular weight, so this is incorrect. They lack a protein coat, they are smaller than viruses, that is correct. So viruses and prions are similar in size, but viroids are smaller. Okay, remember this. Correct option here is B. 72% have answered correctly. Sanya, Ram and Lakshmana. Wow. No, that's not Lakshmana. I thought Ram and Lakshmana both are there. This is Lasmana. Alright. So correct answer is option B. Here is the next question. In today's lecture, Ansh, we are going to cover all of the 11th grade chapters, 10 chapters. We are still in the second chapter. We have 8 more chapters left. But in some chapters, moving forward, not many questions are asked. So, I'm getting a mixture of A, C, D and B. The term contagium vivum fluidum was coined by Bejanic, yes. Mosaic disease in tobacco and AIDS in humans are caused by viruses, yes. The viroids were discovered by 𝐄𝐍𝐆𝐋𝐈𝐒𝐇. No. It was discovered by Diner, so this is incorrect. Stanley showed that viruses could be crystallized, this is correct. So correct answer to this question is option C. Only 21% have answered. Kaushik, Aanchal and Harshit, this is a question from 2015. So Ivanovsky was the person who identified that the causative organism for tobacco mosaic disease in tobacco, that was very smaller and it could pass through bacteria proof filters. Alright. Pagenik, he is the person who called that infective fluid as contagium vivum fluidum. Stanley is the person who have first crystallized the virus. Okay. Oh, here is a question. We just discussed this. Lisa, this class will go on until at least 3 o'clock, I think. If your name is not showing, Geetesh, then at the top of the chat box, there is a pinned comment. There is a link there, Unity link. You have to click on that and enroll and then participate. It will reflect in the poll here. What did he discover? Free infectious RNA. He discovered viroids, but they have not directly given viroids. Free infectious RNA. So option D is the correct answer. 83% have answered correctly. Aanchal, Lisa and Soni are in the top 3 spots. Now let's move on and take a look at our next question. We are solving last almost 10 papers of all 11th grade chapters so that you get benefited. If you find that it's long and it's draining your energy, you can choose not to watch it. What are the different parts? A is the head, right? There's only one option with A as the head. So option C is the correct answer. B, B is the sheath, C is the collar, D is the tail fibers. Option C is the correct answer. 87% have answered correctly. Lisa, Geetesh. See, Geetesh, your name is here finally. And Ansh are in the top 3 spots. This is the diagram that is given to you in NCERT. With that, we have finished the questions from the chapter Biological Classification. Let's take a look at the leaderboard. Ram is on top of the list. He's answered 30 out of 34 questions correctly. Congratulations, Ram. That's a great, that just shows how well you prepared. Ansh is in the second spot and Suresh is in the third spot. Quickly behind we have Rohit and Anjali and Mayuri, Vishal, Abhishek, Jyoti and Tanmayi in the top 10. Congratulations to everyone participating. There are a lot more questions coming up. Make sure you answer correctly so that you will rank up in your leaderboard. Alright, so the next chapter that we have right now is the chapter Plant Kingdom. In Plant Kingdom, the last part where you had all these different types of life cycles, haplontic, diplontic, haplodiplontink, that part is deleted. It's okay, Lisa, we have a lot more questions. You can answer all of those and you can rank up in the leaderboard. So, yeah, so that part is deleted. Everything else is there in this chapter. So, on an average, you can expect around 3 to 4 questions from this chapter in your NEET exam. Here is the first question. Which of the following is incorrectly matched? Which of them is incorrectly matched? Ectocarpus is a brown alga, Eulothrix is a green alga, Porphyra is red alga, Volvox is green alga. In brown algae, fucoxanthin is the pigment responsible, so this is correctly matched. In green algae, mannitol is not the reserve food, it's simple starch, so this is incorrect. In Porphyra, Floridian starch is the reserve food material, that is correct. In Volvox, it is starch, this is also correct. What is incorrect is option B. 82% have answered correctly. Lisa, Aegis and Shri are in the top 3 spots. Congratulations. So here is the mnemonic that I want you to remember for all the green algae. The different green algae examples are Euderina, Eulothrix, Chlorella, Volvox, Cara, Chlamydomonas and Spirogyra. So if you remember this image and this statement, you will remember which are your green algae, right? So your is for Euderina, Ultra is for Eulothrix, Clean is Chlorella, Volvo is Volvox, Car is Cara, Calms is Chlamydomonas, Spirit is Spirogyra. Your Ultra, Clean, Volvo, Car, Calms, My Spirit, if you remember this, you will remember all the green alga. Likewise, there is a mnemonic for brown algae. Sir dictates extremely lame facts. Sir is for sargassum, dictates is for dictyota. Extremely is for ectocarpus, lame is for laminaria and facts is for fucus. Likewise, for red algae, this is the mnemonic. Poly prepared grass jelly, poly is for Polysiphonia, prepared is for Porphyra, grass is for Gracilaria, jelly is for Gelidium. Okay, so I hope you will remember with the help of this mnemonic, which I have also given in your 50-day NEET challenge. Lisa, not everybody here understands and speaks Hindi, so it might sound like French and Latin to them. Since this is an entirely English, pure English channel, I decided to make something in English and I came up with this. Hydrocolloid carrageenan is obtained from chlorophyce, phaeophyce, phaeophyce, rhodophyce, rhodophyce only, phaeophyce only. It is in rhodophyce only. So the correct answer to this question is option C. 83% have answered correctly. Lisa, Anchal and Tanmay are in the top 3 spots. This is a question from 2022. You can see how they are so direct, right? Wait, what happened? Which of the following algae produce carrageenan? Blue green algae, brown algae, red algae. They can ask you questions like this or give you examples of each of these algal groups. The correct answer to this question is red algae. We just solved it in the previous question. So option D is the correct answer here. 93% have answered correctly. Sweetie, Lisa and Anjali are in the top 3 spots. Here is our next question. Which of the following algae contains mannitol as reserve food? Everybody knows that mannitol is the reserve food in brown algae, right? In brown algae. Among the given options, which is the brown alga? Eulothrix is green, Ectocarpus is brown, Gracilaria is red, Volvox is green. So correct answer here is option B. 79% have answered correctly. Archel, Lisa and Sweetie are in the top 3 spots. This is a question from 2021. Here is our next question. Floridian starch has a structure that is similar to. This is a question from 2020. Starch and cellulose, amylopectin and glycogen, mannitol and algin, laminarin and cellulose. The correct answer here is option B, amylopectin and glycogen. This is a statement given to you directly in NCERT. If you have read it thoroughly, you should answer it, very easily. Ram, Anchal Singh and Sai Mona are in the top 3 spots. Hi Sai Mona, welcome to the lecture. Here is our next question. Which of the following pairs is of unicellular algae? Which is unicellular? Laminaria and Sargassum both are multicellular. Gelidium, Gracilaria are multicellular. Volvox is colonial. Anabina, no. Chlorella and Spirulina, they are unicellular. Correct answer to this question is option D. 90% have answered correctly. We have Sweetie, Lisa and Mona in the top 3 spots. Here is our next question. I think I already gave out the answer to this in the previous question. An example of colonial alga. Volvox, Eulothrix, Spirogyra, Chlorella. Chlorella is unicellular. Eulothrix and Spirogyra, they are multicellular and filamentous. Volvox is colonial. So the correct answer is option A. Everyone's answered correctly. Lisa, Anchal and Sweetie are in the top 3 spots. Here I've added a slide to, I've basically given the examples of different forms of algae here. Alright, now let's move on. Here is our next question. Which one of the following statements is wrong? Geetesh, it will be there in the poll. If you have not answered in the top 3, I'm not scrolling beyond that. After each chapter, I will display the entire leaderboard, but for every question, I will just show the top 3 spots. Okay? Which one of the following statements is wrong? Algae increase the level of dissolved oxygen in the immediate environment. Algin is obtained from red algae and carrageenan from brown algae. No, this is ultified. Algin is obtained from brown algae and carrageenan is obtained from red algae. This statement is incorrect. Agar-agar is obtained from Gelidium and Gracilaria. Laminaria and Sargassum are used as food. Both of these statements are correct. The wrong statement is option B. So the correct answer to this question is option B. 91% have answered correctly. Lisa, Geetesh and Soni are in the top 3 spots. See, Geetesh, did you see your name was there? Male gametes are flagellated in. Yes, it is 𝐄𝐍𝐆𝐋𝐈�𝐄. When you learn about the different ways in which reproduction happens in algae, you know there is asexual reproduction, vegetative reproduction and sexual reproduction. Sexual reproduction happens by the formation and fusion of gametes. Over here, they have asked, what was the question again? Male gametes are flagellated in. Flagellated in 𝐄𝐍𝐆𝐋, but Spirogyra is non-flagellated, no? Ectocarpus has flagellated gametes. They have 2 flagella. Spirogyra is non-flagellated. Polysiphonia, there is no motile cells at all. Anabina, cyanobacteria, no. I think it's Ectocarpus. However, I'm not very sure. Yeah, I think it's A, but I see a lot of you have answered it as B. Lisa, Mona and Rishikesh have answered correctly. You've learnt, know how in brown algae you have flagellated pyriform gametes that are unequal in length. Yeah, right. So here is our next question. It is an assertion and reason type of question. I think one of you had asked for assertion and reason type of question. Yeah, but Geetesh, 𝐄𝐍𝐆𝐋 is not one of the options, no? This was the latest 2023 paper. The first stage of gametophyte in the life cycle of a moss is protonema stage. Protonema develop directly from spores produced in the capsule. Both assertion and reason are correct, and reason is the correct explanation for assertion. Option C is the correct answer. 100% of you have answered correctly. Sanya, Soni and Lisa are in the top 3 spots. So this is basically how the life cycle is in mosses. In the gametophyte, you have Antheridium archegonium. Antheridium produces the male gametes. Archegonium, within it there is a egg which is the female gamete. Antheridium swims in a film of water, comes and reaches the neck of the archegonium, fertilizes the egg and forms the zygote. The zygote develops into the sporophyte. The sporophyte is dependent on the gametophyte. Sporophyte has 3 parts: foot, seta, capsule. Within the capsule, spores are produced. These spores are released. Once the spore germinates, the first phase or the first stage in the gametophytic phase is the protonema. It is filamentous. Protonema directly germinates from the spore. From the secondary branches of the protonema, the leafy gametophyte will form. Okay, so over here the first statement was, the assertion statement was, the first stage of gametophyte in the life cycle of a moss is protonema stage. That is true. The reason is because it directly develops from the spore produced in the capsule. So correct answer is option C. I hope this is clear. I'm sure it is clear. All of you had answered correctly. Here is our next question. 12th syllabus we are not doing today, Lisa. That will be done on 24th, I think, not today. Gemme are present in some liverworts, mosses, pteridophytes, some gymnosperms. The correct answer is option A. It is present in some liverworts. So this is what it looks like. cute little things, very pretty. What are you looking at here? These cup-like things are called gemma cups. All these biscuit-like tiny cookie-like things, they are your gemme. They are multicellular, asexual bud-like structures produced within these cups. When these are released, when they find a suitable substratum, they will give rise to a new thallus. So correct answer over here is some liverworts. Okay. 91% had answered correctly. Tejaswini, Sanya and Shri are in the top 3 spots. Here is our next question. Which one is wrongly matched? I will not have another class in this series. I will be dealing with the first year syllabus, 12th grade syllabus. Chaitra ma'am will be handling. Which one is wrongly matched? Polysiphonia, remember it is a red alga. Red algae do not have any flagellated cells in its life cycle. So its gametes are not flagellated. It is incorrect. In brown algae, you find biflagellated zoospores that are laterally, that arise from the side laterally. Gemma cups are seen in liverworts like Mercantia. Chlorella is a unicellular organism. All of these are correct. What is wrongly matched is option A. This is a question from 2018. Only 50% have answered correctly. Lisa, Tejaswini and Mona are in the top 3 spots. Let's move on to our next question. Which of the following is responsible for peat formation? You learn about this in bryophytes, economic importance or ecological significance of bryophytes. Which of the following is responsible for peat formation? It is option D, sphagnum. Right? It is also called peat moss. So correct answer here is option D. 93% of you have answered correctly. Sweetie, Anjali and Tejaswini are in the top 3 spots. Tejaswini has been consistently answering correctly and quickly. Congratulations, Tejamma. Here is our next question. Identify the pair of heterosporous pteridophytes among the following. Heterosporous pteridophytes. This is a question from 2023, last year. Very important question. Psilotum and Salvinia, Equisetum and Salvinia, Lycopodium and Selaginella, Selaginella and Salvinia. The correct answer to this question is option D. They produce 2 types of spores. Microspores that are male spores and megaspores that are female spores. The correct answer here is option D. Lisa, Anchal and Ram are in the top 3 spots. Let's move on to our next question. Lisa, you need me. That's very sweet. But you know what? My son has summer vacation right now. So we are going on a vacation. From next week, I won't be available for an entire week. I'm going to Khasi Hills and Jaintia Hills and all of those places that you've learnt about in biodiversity and conservation. Sacred groves. Genera like Selaginella and Salvinia produce 2 kinds of spores. Such plants are known as? They are said to be heterosporous because they produce 2 different types of spores. Sporous obviously relates to spores. Hetero means more than one. So correct answer is option A. 58% have answered correctly. This was a very easy question. I don't know why so many of you have made mistakes. Sanya, Only Studies and Ram are in the top 3 spot. Only Studies, may I know your real name please? Option A is the correct answer. Here is our next question. Strobili cones are found in Terrace, Salvinia, Mercantia, Equisetum. This is free series, no? Koteshwaramma. This is free series only. Arshin, okay. Strobili are found in ecosystem. What are strobili? 83% have answered correctly. We have Sweetie, Anjali and Lisa in the top 3 spots. You know, in some plants, you have specialized leaves that bear sporangia. We call them sporophylls. Sometimes these sporophylls compactly arrange spirally along an axis and form compact structures called cones or strobili. So that is found in ecosystem. That is your horsetail plant. So the correct answer here is option D. Yeah, thank you, Lisa. Next question. In bryophytes and pteridophytes, transport of male gametes requires birds, water, wind, insects. They need water. Right? They need water for fertilization. So the correct answer to this question is option B. 91% have answered correctly. Tejaswini, Sanya and Manasa are in the top 3 spots. Here let's take a look at our next question. The plant body is not thalloid in Sphagnum, Salvinia, Mercantia, Funaria. Where is it not thalloid? What is thalloid? Thalloid is when the plant body is not differentiated into root, stem and leaves. Then we call it thalloid, right? So in bryophytes, Sphagnum, Mercantia, Funaria, it is thalloid. In pteridophytes, you have true root, stem and leaves. Salvinia is not thalloid. So the correct answer here is option B. 61%, 62% have answered correctly. Prince and Salama and only studies, Arshin, have answered correctly very quickly. In the top 3 spot, we have them. Next question is an assertion and reason type of question. Assertion statement reads, in gymnosperms, the pollen grains are released from the microsporangium and are carried by air currents. And the reason that is given here is, air currents carry the pollen grains to the mouth of the archegonia, where the male gametes are discharged and the pollen tube is not formed. Please read carefully, please read carefully. Is the pollen tube formed or not formed? It is formed. Assertion statement is true, reason is false. Correct answer is option A. Only 14% have answered correctly. There's just 3 of you. Sanya, Salama and Tejamma have answered correctly. This was in 2023. You can see how after the microspores are released, the pollen grains are released, it will form a pollen tube. And after the pollen tube has formed, the male gametes will be discharged inside the archegonia. So do not get confused. Please read the questions carefully. Anjali is doing a thumbs down. I don't know what for. Alright, let's move on and take a look at our next question. Which of the following plants is monoecious? Most students get confused between what is monoecious, unisexual, bisexual, dioecious and all of that. So monoecious and dioecious are with respect to how many different types of plants you have. Mono means one. So monoecious plants are one species will have only one type of plant. In the same plant you will have male and female. Dioecious means you have 2 different types of plants. One plant will have male flowers, the other plant will have female flowers or reproductive parts, whatever, or sex organs. Okay. So in the question they've asked which of the following is monoecious, meaning which one has male and female reproductive structures in the same plant. You can find it in Cara, Cycas, Papaya, Mercantia. All of them are dioecious species. So correct answer here is option C. Jyoti, Tejaswini only studies Arshin in the top 3 spots. Here is what Cara plant looks like. It's a green alga. You can see the male sex organ Antheridium and the female Oogonium both present on the same plant. So it is monoecious. Here is our next question. Let's see how many of you will answer this correctly. This is a question from 2018. Which of the following statements is correct? Which of them is correct? Ovules are not enclosed by ovary wall in gymnosperms. Yes. Selaginella is heterosporous while Salvinia is homosporous. No, Salvinia is not homosporous. It is also heterosporous. Horsetails are gymnosperms. No, horsetails are not gymnosperms. They are pteridophytes. Stems are usually unbranched in both Cycas and Cedrus. In Cycas it is unbranched. In Cedrus it is branched. So this statement is incorrect. We are asked to identify which is the correct statement. So the correct answer to this question is option A. 95% have answered correctly. Tejaswini, Jyoti and Soni in the top 3 spots. Here's what a cedrus plant looks like. You can see how it is so well branched. This is what Cycas is. You may have seen these as avenue trees or avenue plants near your signals and circles in your city. So this is a Cycas plant. It is not branched. So correct answer here is option A. This is our next question. Why do you want to think of anything else? Lisa, why not MBBS? Are you doubting yourself? Do you think you can't do it? Lisa has a Cycas plant in your house. Is it a male plant or a female plant? If it produces like a female cone, it will stink like so bad. Select the mismatch. Cycas is dioecious. Salvinia is heterosporous. Equisetum is homosporous. Pinus is not dioecious. Pinus is monoecious. This is the mismatch. A question from 2017. Option D is the correct answer. 70% have answered correctly. Tejaswini, Sai and Rudra have answered correctly very quickly. They are in the top 3 spots. Rudra is a very nice name. Very unusual. Nice. Conifers are adapted to tolerate extreme environmental conditions because of broad, hardy leaves, superficial stomata, thick cuticle, presence of vessels. They have a thick cuticle. Their leaves are not broad. They are very narrow, needle-like. Stomata is not superficial. It is sunken to reduce the rate of transpiration. Presence of vessels. No, they do not have vessels. Tracheids are mostly used to conduct water. So correct answer here is option C. In the top 3 spots, we have Aanchal, Tejaswini and Arshin. 87% of you all have answered correctly. Next question. Select the correct statement. Sequoia is one of the tallest trees. The leaves of gymnosperms are not well adapted to extremes of climate. No, they are well adapted. Gymnosperms are both homosporous and heterosporous. No, they are exclusively heterosporous. Every gymnosperm will produce 2 types of spores. Salvinia, Ginkgo, Pinus are all gymnosperms. Salvinia is a pteridophyte. So correct answer here is option A. Everyone's answered correctly. Tejaswini, Lisa and Uttej are in the top 3 spots. Here is our next question. In which of the following gametophyte is not independent, free living? Gametophyte is not independent or free living. Pteris is a pteridophyte. Funaria and Marchantia, these are bryophytes. In both bryophytes and pteridophytes, gametophyte have independent existence. Pinus is a gymnosperm. In gymnosperms and angiosperms, you do not have independent existence for gametophytes. So correct answer to this question is option B. 79% have answered correctly. Lisa, PMS and Hima are in the top 3 spots. Okay, with that we are done with the questions from plant kingdom. Let's take a look at our leaderboard. Ram is still on top of the list. He's answered 52 out of 59 questions correctly. Then we have Suresh, and then we have Anjali, and then we have Rohit, then Vishal, Tejaswini, Arshin, Tanmay, Mayuri, Soni, Lisa, PMS, Ansh, Anchal, Sweetie, Abhishek, Swatika, Faumei, I think that's how it's pronounced. Then we have Sai, Mona, Jyoti, Harshit, Sadhana, and the rest of you all. Congratulations to everyone participating and consistently answering. Yeah, Rohit has attempted 61 questions, which is the highest. Congratulations to everyone. Let's move on to our next chapter, which is morphology of flowering plants. In morphology of flowering plants, oh, Shalini, SK Creations, your name is Shalini too? Nice. PMS's name is Anantapriya. What do you want to do in biotech or microbiology, Lisa? What exactly do you want to do? Do you want to pursue research or what interests you? Have you thought about it? Shri, your name will be there, but it's probably somewhere. Let me check. Yeah, so your name is here, Shri. 33. Sometimes some questions the poll will not pick up, so don't rely on this and judge yourself too much. As long as you know how many questions you've answered correctly, it's fine. So in morphology of flowering plants, we have morphology of root, stem and leaves, and then we have adaptations, and we have inflorescence, flower, fruit, seed, all of that. And along with that you have to study about the families. Earlier you had 3 families. I mean in NCERT you have 3 families: Fabaceae, Solanaceae and Liliaceae, 2 dicot families and 1 monocot family. But right now in the reduced NTA syllabus for NEET 2024, newer families have added: Malvaceae, Cruciferae, Compositae, Graminae. All of these are new families. Leguminosae is your Fabaceae itself. Since we are taking PYQs, you will not have questions related to the new families in today's session. If you want questions and concepts related to new families, I want you to refer to the 50-day NEET challenge chapter morphology of flowering plants. Over there we have discussed, Chaitra ma'am has in detail explained the features of the newly added families as well, and also solved the relevant MCQs related to those families. We are not doing that today because today's agenda is to solve PYQs, and in the previous years those chapters were not included. Okay, all right. Now let's move on. From this chapter you can expect around 3 to 4 questions considering the weightage in the past few years. So let's get started with the questions. In the rationalized syllabus, modifications are deleted, but in your NTA NEET syllabus, modifications are still there. The roots that originate from the base of the stem are fibrous primary prop lateral roots, In fibrous roots also, the radicle will elongate and form a primary root, but that is short-lived, it's ephemeral. After that, roots will arise from the base of the stem. They are called fibrous roots. So correct answer here is option A. 95% of you all have answered correctly. Thogni, Jyoti and Ram are in the top 3 spots. Here is our next question. Root hair developed from the region of. Root hair developed from the region of. You know, in the root, you have different regions. You have a root cap, you have the meristematic region, region of elongation, region of maturation. Which region exactly does the root hair arise from? It arises from the region of maturation. So the correct answer here is option D. These root hair will increase the surface area that's available for absorption. 71% of you have answered correctly. Jyoti, Lisa and Hima are in the top 3 spots. Congratulations guys. Here is our next question. Sweet potato is a modified. Is sweet potato a modified stem or root? What is it? It is a type of modified root, more specifically adventitious root. So the correct answer here is option B. 57% of you have answered correctly. Sonali, Jyoti and Samiraj are in the top 3 spots. This is a question from 2018. Here you have a really lengthy statement based question. Identify the correct set of statements. The leaflets are modified into pointed hard thorns in citrus and bougainvillea. It is not the leaflets that are modified, it is the axillary buds that are modified. That makes this statement incorrect. Axillary buds form slender and spirally coiled tendrils in cucumber and pumpkin. That is true. Stem is flattened and fleshy in Opuntia and is modified to perform the function of leaves, which is photosynthesis. That is true. Rhizophora shows vertically upward growing roots that help to get oxygen for respiration. Yes, that is true. Subaerially growing stems in grasses and strawberry help in vegetative propagation. That also is correct. So the correct statements here are B, C, D and E, which is option C. Not bad, 94% have answered correctly. Shiva, Tejaswini and Sadhana are in the top 3 spots. Here's what the citrus and bougainvillea thorns look like. If this is your leaf, if this is the stem and this is the leaf, this is the axil. The buds that are present at the axil is what will modify into thorns. It's not the leaflets or the leaves, right? So here you can see how the axillary buds, this is the leaf, this is the axil. The axillary bud itself is modified into this tendril. You can see that in cucumber, pumpkin, okay, grapevine and all of those. And in Opuntia, stem will take up the function of the leaf. It will perform photosynthesis. It will become flat and green. So that also was a correct statement. Rhizophora, you can see how negatively geotropic roots are formed. They will jut out of the surface of the water and help in exchange of gases. They are called pneumatophores. That is also true. Grasses and strawberry, subaerial growth of stem will help in vegetative propagation. That was also true, right? Now let's take a look at our next question. Bougainvillea thorns are the modifications of stem, right? More specifically, it is the axillary bud that modifies into the stem. Axillary bud is a part of the stem. It's considered as a part of the stem. It is present at the junction or the angle between the stem and the leaf, but still it's considered as a part of the stem. So correct answer here is option B. 95% have answered correctly. Sweetie, Vishal and Soni in the top 3 spots. Here is our next question. Which of the following is not a stem modification? Which of the following is not a stem modification? Tendrils of cucumber is axillary bud, that is a stem modification. Flattened structures of Opuntia is a stem modification. Pitcher of Nepenthes is a leaf modification. Thorns of citrus also is a stem modification. So which is not a stem modification? It's the pitcher of Nepenthes, which is a leaf modification. So correct answer is option C. 82% of you have answered correctly. Ram, Tejaswini and Sameer Raj are in the top 3 spots. Here's what a pitcher plant looks like. The leaves are modified to form a pitcher and they are insectivorous plants. This pitcher will have a slippery surface. It is used to trap insects. Okay, so yeah. Here is our next question. Stems modified into flat green organs performing the functions of leaves are known as. What do we call them? This was in 2016. What it's called as is not mentioned in your textbooks, but yeah. They are called phylloclades. Cladodes are like phylloclades, but they're just generally just one node in length, one internode in length. Phyllodes are when the petiole takes up the function of photosynthesis, as in the case of Australian acacia. So the correct answer here is option A, phylloclade. 93% have answered correctly. Sameer Raj, Priya and Tejaswini in the top 3. Tejaswini also has been very consistent in answering correctly and quickly. Congrats Tejaswini. Here is our next question. Leaves become modified into spines. In which of the following plants? In onion, in silk cotton, in opuntia, in pea plant, in which one? Leaves become modified into spines in opuntia. Opuntia is our cactus plant. Why it gets modified into spines is because to reduce the rate of water loss by transpiration, because these plants live in the desert. If the leaves are entire, more stomata, more transpiration. To reduce that, the leaves are reduced into spines. Such a beautiful adaptation. Option C is the correct answer. Sameer Raj, Arshin and Ram in the top 3 spots. Here is an assertion and reason type of question. Let's take a quick look at it. Read carefully and answer. A flower is defined as a modified shoot wherein the shoot apical meristem changes to floral meristem. Internode of the shoot gets condensed to produce different floral appendages laterally at successive nodes instead of leaves. Correct answer is option C. Both assertion and reason are true, and reason is the correct explanation of assertion. If you consider like a normal shoot, what will you have? You will have a stem, and in the stem you will have these nodes. At the nodes you will have leaves, right? When the stem gets modified, it will produce flowers. So what happens is, at each node, instead of producing normal leaves, it will produce modified leaves. What are these modified leaves? Let's see. At one node, it will produce the sepals. At the next node, it will produce the petals. At the next successive node, it will produce stamen. And then in the next node, it will produce your carpel. So this is one thing that happens. These are your nodes, the regions between your nodes. This is your internode. Firstly, leaves are modified into different parts of the flower. Next, the internodal region gets super condensed. Internodal length becomes very short, and it will appear like all of these parts are arising from the same point, and it gets modified into this. Wow, what have I done? Stop. This is modified and condensed shoot. So assertion is true, reason is also true, and the reason correctly explains the assertion. So correct answer to this question is option C. 63% of you have answered correctly. Soni, Jyoti and Sanya are in the top 3 spots. This was a question from our latest 2023 NEET exam. Here is our next question. In a CMOS inflorescence, the main axis. I called Harry Priya. I don't know. Hi Nitish, good afternoon. In CMOS inflorescence, it does not have unlimited growth. It does not bear a solitary single flower. It has unlimited growth but lateral branches in a flower. No, it does not have unlimited growth. It terminates in a flower. Yes, this is the correct statement. Correct answer to this question is option D. 65% of you have answered correctly. Jyoti, Lisa and Tejaswini in the top 3 spots. Here's what the different types of inflorescence are. This is your racemose inflorescence. In racemose inflorescence is where you will find indefinite or unlimited growth, because over here the floral axis does not terminate in a flower. You have the older flowers present towards the base and younger flowers present towards the tip. So such a type of arrangement is called acropetal arrangement. You can see that in case of Gulmohar. CMOS inflorescence is when the floral axis terminates in a flower. Oldest flower is at the tip, younger ones are present below it. Such a type of arrangement is called basipetal succession or basipetal order. Example for that is jasmine. Okay, I hope you've understood this question correctly. Here is our next question. Yeah. Axile placentation is observed in. Option B is the correct answer. China rose, petunia and lemon. Option B is the correct answer. 86% have answered correctly. Sameer, Raj, Tejaswini, Jyoti in the top 3. These are the different types of placentation. I've added examples so that you can revise very quickly. In axial placentation, as asked in the question, you have like a central axis sort of a thing. It is a multilocular ovary. To this axis, ovules will be attached. That is axial placentation. You can see that in China rose. You can see that in tomato. You can see that in lemon. But if you see in the option, it is given China rose, lemon and petunia. Petunia is not mentioned as it is over here in your NCERT example, but in the family Solanaceae, tomato and petunia, both of those are examples. So that is how you should relate, and the correct answer here is option B. Alright, now let's look at this question. Lisa, B is correct. The flowers are zygomorphic in. Zygomorphic means what? It exhibits bilateral symmetry. Mustard is actinomorphic. Gulmohar is zygomorphic. Cassia is zygomorphic. Datura, chili are both actinomorphic. B and C is the correct answer. So option B is the correct answer. Yes, 76% have answered correctly. Tejaswini, Jyoti and Arshin in the top 3 spots. Here's how you can categorize flowers based on symmetry. If you can cut a flower into 2 equal halves in any plane, we call that actinomorphic. Examples for that are mustard, datura and chili. Zygomorphic, in any one vertical plane, you can get 2 equal halves. Pea, gulmohar, bean, cassia are examples. Asymmetric means in any plane you do not get 2 equal halves. Example for that is canna. Okay, I've added this so that It's easy for you to revise. Here is our next question. Radial symmetry is found in the flower of Brassica, Trifolium, Pisum, Cassia. All of these are not radially symmetrical. All of these show bilateral symmetry. Brassica shows radial symmetry. So the correct answer here is option A. 73% have answered correctly. Jyoti, Yuraj and Sadhana are in the top 3 spots. Let's take a look at our next question now. Which one of the following plants shows vexillary aestivation and diadelphous stamen? Colchicum autumnale, Pisum sativum, Allium cepa, Solanum nicarum. Vexillary aestivation is a characteristic feature of your pea family, bean family, Fabaceae. So correct answer here is option B. What exactly is your vexillary aestivation like? You will have a total of 5 petals. You have one like super large petal which is called a standard or vexillum. Then you have the wings or alae, 2 smaller petals that are overlapped by one large petal, and then you have 2 even smaller petals that are fused. This is called keel or carina. So this is your vexillary aestivation. We call this papilionaceous corona. You can see this in the petals of Fabaceae members. So diadelphous stamen is when you have stamen fused. The filaments are fused forming 2 bundles. In Fabaceae members you see 9 stamen forming 1 bundle and 1 stamen present separately. We consider this as 2 bundles. We call this diadelphous stamen. So both of these are seen in Pisum sativum. So the correct answer to this question is option B. 79% have answered correctly. Aanchal, Jyoti and Lisa are in the top 3 spots. Congratulations. Let's take a look at our next question, which is also based on diadelphous stamens. Diadelphous stamens are found in P. I just told you. So the correct answer is option D. 81% have answered correctly. Arshin, Jyoti and Lisa are in the top 3 spots. This was a question from 2021. In China rose or hibiscus, you can see monoadelphous stamen. In citrus, you can see polyadelphous stamen. In pea, it is an example for diadelphous stamen. Here is our next question. Ray florets of sunflower. Ray florets of sunflower. Whenever the flower is epigynous, then the ovary will be inferior. What does epi mean? Epi means above. Gynous means gynoecium. It relates to the gynoecium or your ovary or your female reproductive part. Now this classification of flowers is based on with respect to the ovary or with respect to the gynoecium. Where do the other floral whorls arise from? Do they arise from below the ovary? Do they arise from above the ovary? Do they arise from the same level of the ovary? That is what we need to see here. So the ray florets of sunflower are epigynous. The flowers are epigynous. That means if this is your ovary, the other floral parts arise from above the ovary. If the ovary occupies the lowermost position, we call it the inferior ovary. Alright? So the correct answer to this question is Option A. 83% of you have answered correctly. Sameeraj, Lisa and Tejaswini in the top 3 spots. Would you want to take Rohit? Hi Kamini, good afternoon. TP want to take SS. What does that mean? The ovary is half inferior in plum, rose and peach. So the correct answer is option D. 73% have answered correctly. Jyoti, Tejaswini and Lisa are in the top 3 spots. Right? Alright, let's move on. Placentation in which ovules develop on the inner wall of the ovary or in the peripheral part is free central, basal, axile, parietal. I am getting mostly correct answers. The correct answer to this question is option D, parietal placentation. Tejaswini, Sweetie and Jyoti are in the top 3 spots. Let's take a look at the different types of placentation. Here is what it looks like. This is the wall of the ovary. Seeds or ovules are attached to the peripheral part. So that is a parietal placentation. Next question. Free central placentation is seen in free central placentation. What is free central placentation? It is seen in dianthus and primrose. So correct answer here is option A. 91% have answered correctly. Jyoti, Sameeraj and Sadhana are in the top 3 spots. Here is our next question. Placentation in tomato and lemon is. Oh, I got an ad here. Placentation in tomato and lemon is. Everybody has memorized all of these examples really well. It is showing. The correct answer to this question is option D. It is axile placentation. You can see how frequently questions related to placentation are repeated. So you must be very thorough with the examples. 82% have answered correctly. Jyoti, Sameeraj and Lisa are in the top 3 spots. Okay. Next question. Match the following. Again, based on placentation itself. It's a really important concept. Basal placentation can be seen in marigold, no, sunflower, axial placentation, What do you think is the correct answer? China rose. Option C is the correct answer. 94% have answered correctly. Tejaswini, Jyoti and Lisa in the top 3 spots. Why is this slide here? Never mind. The term polyadelphous is related to. Poly means many, right? Poly means many. Adelphous. This is related to stamens fusing. Like in the stamen you have the filament and you have the anther. Filaments fusing to form bundles is called adelphi. If they are fused to form more than one bundle, then we call it polyadelphous. So this is related to androecium. It is related to stamens. So the correct answer here is option B. 92% have answered correctly. Jyoti, Sweetie and Tejaswini in the top 3. Here is our next question. We've already discussed this in another question. I hope everybody answers this correctly. The standard petal of a papilionaceous corolla is also called. What is papilionaceous corolla? Papilionaceous means butterfly-like. In pea plant or the pea family, Fabaceae, petals resemble butterflies, so they are called papilionaceous corolla. What is the standard petal also called? It's also called vexillum. So the correct answer to this question is option A. 92% have answered correctly. We have Jyoti, Sushant and PMS in the top 3 spots. Let's move on to our next question. Yeah, I already displayed this slide initially also, earlier also. These are the different types of placentation with examples I've added here so that you can revise quickly. Lisa's cracking a joke. C option is Karen now. Keel is the characteristic feature of a flower. Aloe, tomato, tulip, indigofera. Aloe, tulip, they are monocot family. Tomato, Solanaceae, indigofera is Fabaceae. So the correct answer here is keel. Those two small petals that are fused. That is characteristic of indigofera. So the correct answer to this question here is option D. 76% of you will have answered correctly. Yuraj, Ram and Easy Math is on the top 3 spots. Here is our next question. Coconut fruit is it a berry, is it a nut, is it a capsule, is it a drupe? Coconut fruit is a drupe. The outer leathery layer, in a tender coconut, that is your epicarp. Inside you have the fibrous layer, that is your mesocarp. The hard layer inside is your stony endocarp. Coconut is an example for a drupe. Also, mango is an example for drupe. Both of them have one seeds only. So correct answer here is option D. 96% have answered correctly. Jyoti, Lisa and Ram in the top 3 spots. Let's take a look at our next question. Cotyledon of maize grain is called. Maize is a monocot plant. In maize you have only one cotyledon and that one cotyledon is large and shield shaped. What do we call it? We call it the scutellum. Option B is the correct answer. What is plumule? The tip of your epicotyle is called your plumule. What is coleoptile? Coleoptile is the sheath that covers your plumule. What is your coleorhiza? It is the foliar or leafy sheath-like structures that cover your radicle. Right? So the correct answer to your question here is option B. 84% have answered correctly. Sameeraj, Anchal and PMS in the top 3. With that, we have finished the chapter. Morphology of flowering plants. At the top of the leaderboard, Ram has retained his position consistently. He's answered 75 out of 85 questions correctly with an average time of 11 seconds. Congratulations, Ram. Suresh was, I think, in the third spot. The last time we checked the leaderboard, he is now in the second spot. Congratulations to Suresh as well. Then we have Anjali, Tejaswini, Rohit, Soni, Vishal, Lisa, Only Studies, PMS, Anchal Singh, Tanmay, Sadhana, Sai Mona, Sweetie, Mayur, Jyoti, Ansh, Shirisha, Abhishek, Swatika, Shri, and the rest of you all. Lisa has to go. Where are you going, Lisa? Are you hungry? We may not meet again, Lisa, in this class. So let's finish anatomy of flowering plants and then we'll take a little break for lunch if you all are hungry. If you need a break. Oh, hi. You're from Khasi Hills. That's really nice. It must be such a beautiful place to live in, no? Where is Shri going? Everybody is going. Okay. So, we'll take a break after anatomy of flowering plants. We'll be done with like 2 units. We'll be left with 2 more units. We can come back later. Do you want to take a break now? Let's take a break after finishing this chapter, I think. What do you think? Do you want to take a break now? How many of you want a break now? I will conduct a poll. If you want a break, type A. If you do not want a break, then type B. A for break, B for no break. Type A for break, B for no break. If you want a break, you type A. If you don't want a break, type B. Okay, majority of you don't want a break. So let's continue. After this chapter, we'll take like a little break for lunch. In the meantime, tell your parents or your mother or whoever is cooking at home that you'll be taking a break soon and we will take a very short break. I'm not giving you like half an hour, 1 hour to happily have your lunch and come back. So yeah. After anatomy, Shri, we have cell the unit of life, we have cell cycle and cell division, photosynthesis, respiration, plant growth and development. Alright. So in anatomy of flowering plants, we learn about tissues, we learn about tissue systems, we learn about the anatomy of roots, stem and leaves both in monocots and dicots. There used to be a concept called secondary growth earlier, but in this year's NEET syllabus, that portion is deleted. Alright. Kashmir, yes, I want to visit Kashmir. We are planning to go there probably in June, July sometime. Is it a good time to visit Kashmir? When is the best time to visit Kashmir? Okay. Yeah, I'll visit every place to say congratulations to you after you get your NEET results and you secure a seat in a government seat in a medical college. Then I'll come to every place and say congratulations to you. June and July is the best time. Yeah, that is when we are planning. Where can you find notes, Sushant? Sushant, after this class is over, this notes will be available as PDF in the description box. You can go click there. Pushpa the fire. Zoology, I think Vindhya ma'am and Chavi ma'am will be teaching. Okay, Soni, I'll visit Bihar as well sometime. So, anatomy of flowering plants. In the past years, you can see how there are 2 to 4 questions asked. Boring games, what's your real name? Same to you, Ani. I don't know, let me check. Have you registered in Unity? Can I search your names? Huh, you're there. You've answered only 19 questions out of 16. That's why. Oh yeah, Aditya, I remember you used to attend this 50-day challenge, right? Okay, so everyone's ready for Anatomy of Flowering Plants questions? Let me know in the comments. Thumbs up and let me know, is everyone ready? झुकेंगे नहीं. Okay, let's get started, guys. Here is our first question. It is a statement based question. This was asked in 2023. Given below are two statements. What's the first statement? Endarch and exarch are the terms often used for describing the position of secondary xylem in the plant body. Endarch and exarch are the terms often used for describing the position of secondary xylem in the plant body. No, not at all. It is used to describe the primary xylem, specifically protoxylem's position. First statement is incorrect. Exarch condition is the most common feature of the root system. That is true. So statement 1 is incorrect, but statement 2 is true. Option B is the correct answer. 86% have answered correctly. Tejaswini, Sadhana and Ram are in the top 3 spots. Do you have any confusion here? Should I explain what endarch and exarch is, or do you already know it? The first formed xylem in a plant is called the primary xylem. In primary xylem, you have vessels of varying diameters. Some of them will have a smaller diameter, some of them will have a larger diameter. Smaller diameter is protoxylem. The one that has larger diameter is your metaxylem. Depending upon where this protoxylem is facing, you have endarch and exarch. If protoxylem is facing the pith towards the inside, we call it endarch. If the protoxylem is facing the periphery towards the epidermis outside, then we call it exarch. Okay, so endarch you find in roots, exarch you find in stems, both monocot and dicot. Okay, so correct answer here is option B. Let's take a look at our next question now. Phloem in gymnosperms. Phloem in gymnosperms lacks both sieve tubes and companion cells, albuminous cells and sieve cells, sieve tubes only, companion cells only. They lack both sieve tubes and companion cells. Instead of sieve tubes, they have sieve cells. Instead of companion cells, they have albuminous cells. What they lack is both sieve tubes and companion cells. So the correct answer here is option A. 85% of you have answered correctly. Sameer, Raj, Tejaswini and Pushpa have answered correctly very quickly. Pushpa is in the top 3. In Kannada we say late ag bandru, latest agi bandve anta. That means even though you are late, you are very latest. Like Pushpa started watching the session, started attempting the questions only like 5 minutes ago, but he's already in the top, one of the top 3 spots, he or she, whoever. So yeah. Regeneration of damaged growing grass following grazing is largely due to which type of meristem? Lateral meristem, apical meristem, intercalary meristem, secondary meristem. The correct answer is intercalary meristem. Intercalary meristem you usually find in the internodal region between your permanent tissues. So correct answer here is option C. 86% have answered correctly. Tejaswini, Anchal and Sameer Raj are in the top 3 spots. Here is our next question that was asked in Karnataka NEET 2013. Meristematic tissue responsible for increase in girth of tree trunk. Girth means the diameter of tree trunk. Actually, secondary growth is deleted. I should not have picked this question, but anyway. You have different types of meristematic tissues, right? Among these, which one helps in increasing the girth of tree trunk? It happens because of lateral meristem, that is your cambium. So correct answer to this question is option B. 89% of you have answered correctly. Jyoti, Mona and Sameer Raj are in the top 3 spots. Here is our next question. The chief water conducting elements of gymnosperm, water conducting elements of xylem in gymnosperm are. Water conducting elements of xylem in gymnosperms are. Do gymnosperms have vessels at all? Generally, water gets conducted through vessels and tracheids. But vessels are a characteristic feature of angiosperms, if you remember. Angio itself means vessel, right? So, angiosperms are that group of plants which have vessels in their xylem, and in angiosperms, majorly water gets transported through vessels. But gymnosperms, most gymnosperms lack vessels. Over there, transportation of water mostly happens through tracheids. So the correct answer to this question is option D. Wow, 50% have answered correctly. Mona, Easy Math and Tejaswini are in the top 3 spots. Let's take a look at our next question, which is like super lengthy. Read carefully. This is a question from 2022. There are 5 statements given to us in this question. Read the following statements about the vascular bundles. In roots, xylem and phloem in a vascular bundle are arranged in alternate manner along with different radii. Conjoined closed vascular bundles do not possess cambium. In open vascular bundles, cambium is present between xylem and phloem. The vascular bundles of dicotyledonous stem possess endark protoxylem. In monocot root, usually there are more than 6 xylem bundles. Choose the correct answer from the options given below. What do you think is the correct answer? Is any statement wrong here? Is any statement wrong here? In roots, xylem and phloem are arranged in alternate manner along different radii. That is true, right? There we see radial arrangement of vascular bundles. This statement is correct. Conjoined closed vascular bundles do not possess cambium. That is why we call it closed. That is also correct. In open vascular bundles, cambium is present between xylem and phloem. That is also correct. The vascular bundles of dicot stem. In a dicot stem, you know how? It is endark. That is also correct. In monocot root, there are more than 6 xylem bundles. That is also correct. Everything is correct. This was a faulty question asked in NEET 2022 for which they were awarded grace marks. So, why is B not right? In conjoined closed, there is no cambium, right? There is no cambium. It's a correct statement. Why is it wrong? Purnima and Soni. Conjoined is when there is no right option. All of these statements are correct. In the options, they've given either 3 or 4 statements and said only. So none of these options are correct because all the statements given to us here are correct. So, okay, cool. Got it now. Alright. So this is a question for which grace marks was awarded. Now what do I do with this poll? There are no correct answers. Let's just pick A. Here is a match the following type of question. Under list 1, you have characters of some type of tissue or cell. Some type of tissue only. And under list 2, you have different types of tissues listed. You have to match it correctly. Very simple question. You've studied about this in 9th grade only, I think. This was asked in 2021. Cells with active cell division capacity, what do we call them? We call them meristematic tissues, right? So P is paired with 2. Tissue having all cells similar in structure and function. If they are all very similar, then we call it simple permanent tissues. Tissues having different types of cells. Vascular tissues, they are complex permanent tissues. They have 4 different types of cells, each in phloem and xylem. Dead cell walls with highly thickened walls, dead cells with highly thickened walls and narrow lumen, sclerenchyma, sclerites, option 3, not option 3, like S is paired with 3. Correct answer is P2, we have only one option with P2, P2Q4R1S3, which is option B. 93% have answered correctly. Tejaswini, Jyoti and Easy Math are in the top 3 spots. Here is our next question from 2018. Stomata in grass leaf are. Grasses are monocots. In monocots, what is the shape of? Actually, this should be guard cells instead of stomata. Guard cells in grass leaf are dumbbell shaped, kidney shaped, rectangular, barrel shaped. In monocots, it is dumbbell shaped. It is kidney shaped in dicots. Okay, so the correct answer here is option A. 78% have answered correctly. Sweetie, Easy Math and Sameer Raj are in the top 3 spots. I wonder why some of you made mistakes in this question. It was such a simple question, right? It was not simple. Aditya? No? Alright. Let's move on. Take a look at our next question. Specialized epidermal cells surrounding the guard cells are called. You cannot afford to be making silly mistakes now. Please read carefully and answer. Specialized epidermal cells surrounding the guard cells are called bulliform cells, lenticells, complementary cells, subsidiary cells. The correct answer to this question is option D. Only 65% have answered correctly. Tejaswini, Yuvraj and Yuraj and Jyoti are in the top 3 spots. This is a question from NEET 2016. NEET 2016 paper was thought to be one of the toughest NEET papers. A lot of questions were asked out of NCERT. Here is our next question. Vascular bundles in monocots or monocotyledons are considered closed because, why are we considering vascular bundles in monocots closed? There are no vessels with perforations. Monocots are still angiosperms. Vessels will be present. Xylem is surrounded all around by phloem. No. A bundle sheath surrounds each bundle. Okay, no. That's not why we call it closed. Why do we call it closed? Because cambium is absent. So why should we call it closed? It is closed for secondary growth. Closed for secondary growth. Now in plants, in some plants, like if you take a mango plant, you sow the seed and then you get a little plant, and that plant will grow up. As it grows, it will increase in height, increase in length as well as in girth, right? So increase in length is called primary growth, increase in girth is called secondary growth. Increase in girth or secondary growth happens because of the presence of a type of lateral meristem called cambium. In monocots, that cambium is not present. That type of lateral meristem is not present. As a result, you do not see like a sugarcane plant growing into a huge trunk or a bamboo plant growing into a huge trunk. Over there, secondary growth is not seen. Since cambium is absent, that is closed for secondary growth. So the correct answer to this question is option D. I hope all of you have understood this correctly. 86% of you have answered correctly. How do I even read this name? He's answered like really quickly in less than 1 second, he or she. Is this some made up name? Then we have Sai and Jyoti in the next two spots. Odugu. Where are you from? Okay. Now let's move on and take a look at our next question. Ground tissue includes I think they are from northeast or somewhere maybe, because the name is not very familiar or popular in South India at least. Ground tissue includes, what does ground tissue include? All tissues external to endodermis, all tissues except epidermis and vascular bundles, epidermis and cortex, all tissues internal to endodermis. Correct answer is option B. Ground tissue includes everything except for your epidermal tissue and your vascular tissue. So the correct answer here is option B. 82% have answered correctly. Samiraj, Tejaswini and Sweetie are in the top 3 spots. Some vascular bundles are described as open because these are surrounded by pericycle but no endodermis. Pericycle and no endodermis? Not at all. Capable of producing secondary xylem and phloem. Possesses conjunctive tissue between xylem and phloem. Are not surrounded by pericycle. They are capable of producing secondary xylem and phloem. Secondary xylem and phloem are produced because of the activity of a type of lateral meristem which we call cambium. So if cambium is producing more and more secondary xylem and phloem, the stem or even the root in case of dicots will increase in girth. Okay. So the correct answer here is option B. 78% have answered correctly. Tejaswini, Jyoti and Easy Math are in the top 3 spots. Here is our next question. Bharat AYS Bharat Payal D is saying D from that time. I don't know if it's D or if it's D. In barley stem, vascular bundles are, barley is an example for monocot. In monocots, vascular bundles, especially in monocot stems, vascular bundles are closed and they are scattered. Closed means there is no cambium. Scattered, you know, if you look at it under the microscope, it looks like skulls. If you may have seen in schools, in your practical classes, it looks very pretty, like so many skulls are scattered. Option A is the correct answer. 50% have answered correctly. Tejaswini, Sweetie and Sadhana in the top 3 spots. Here is another lengthy question that is statement based. Let's see how many of you will answer this correctly. Didi is like akka, no? You know, I have some Tamil friends in Tamil. D is like bro. You say what bro or something like that. In Tamil, we say what D. The transverse section of a plant shows the following anatomical features. Large number of scattered vascular bundles surrounded by bundle sheath. Okay, when we say scattered, we should already get an idea there. Large conspicuous parenchymatous ground tissue. Correct. Vascular bundles are conjoined and closed. Okay. Phloem parenchyma is absent. Okay. So this is obviously our monocot stem. Correct answer here is option A. 88% have answered correctly. Samir Raj, Easy Math and Yuraj are in the top 3 spots. Samir Raj has been consistently answering correctly since the last few questions. Congratulations, Samir. Here is our next question. This is from NEET 2019. Grass leaves curl inwards during very dry weather. Select the most appropriate reason from the following. Tylosis in vessels, closure of stomata, flaccidity of bulliform cells, shrinkage of air spaces in spongy mesophyll. Grasses are monocots. In monocots, what happens? In your adaxial epidermis, very close to your vascular bundles, you have specialized cells that are in the epidermis. They are epidermal cells, but they look very different from the other epidermal cells. They are much larger in size, they are colorless, they are empty. So what happens basically when the sunlight is too much or the temperature is very high or the weather is very dry, when the leaf is fully spread out like this, it's exposed more to the, it's more exposed. So more stomata are exposed, more water loss will happen. If the weather is already dry, then the plant will lose a lot of water and transpiration. So in order to prevent or reduce that rate of transpiration, these bulliform cells that are usually filled with water will lose water and become flaccid. When they become flaccid, the leaf will roll. When the leaf rolls, the area exposed will be reduce. As a result, rate of transpiration also will reduce. So the correct answer to this question is option C. 84% have answered correctly. Tejaswini, Sweetie and Anjali are in the top 3 spots. Lawrence, Tihar Jela, what all names you come up with, man. Here is our next question. Casparian strips occur in epidermis, pericycle, cortex, endodermis. Casparian strips are deposition of suberin in the radial as well as tangential walls of endodermis, both in monocot and dicot roots. Okay, so that is called Casparian strips. It is found in endodermis. Correct answer to this question is option D. 65% have answered correctly. Tejaswini, Anvay and Vani have answered correctly. They are in the top 3 spots. Next question, very simple from NEET Phase 2 2016. Cortex is the region found between epidermis and stele, pericycle and endodermis, endodermis and pith, endodermis and vascular bundle. What do you think is the correct answer? Correct answer is option A, in between the epidermis and stele, which starts with the pericycle. Everything else is your cortex. Correct answer is option A. Only 17%. 18% have answered correctly. Jyoti, Sweetie and Sanya. Top 3. Here is our next question. Pushpa, are you saying you had left the class till now, or are you saying that you want to leave the class now? Are you asking for permission, or are you informing that you were not there? Very polite set of children. I really like this batch. Sameer Raj, how can Hyderabad be any answer here? Who asked you anything about Hyderabad? You are given a fairly old piece of dicot stem and a dicot root. Which of the following anatomical structures will we use to distinguish between the two? How do you differentiate between a dicot stem and a dicot root? Both of them are fairly old, they are saying. What can you use? Actually, these questions are pretty straightforward, not really very tricky in my opinion. Yes, that's what I said. Everyone is polite in this batch. Yeah. So, protoxylem in dicot stem. In stem it is endarch, in root it is exarch. So, correct answer here is option C. 63% have answered correctly. Sameer, Raj, Tejaswini and Sadhana are in the top 3 spots. And with this, we have finished the important MCQs from the chapter Anatomy of Flowering Plants. At the top of the list, Ram is still there, secured his safe space, safe place. Followed by that, we have Suresh. Again, he was in the second place the last time we checked as well. Then we have Tejaswini and Anjali, Rohit, Soni, Sadhana, Vishal, Aanchal, Lisa, PMS, only studies, Sai Mona, Tanmay, Jyoti, Sweetie, Mayuri, Ansh, Easy Math, Shirisha, Aditya, Shri, Sameer Raj, Sanya, Abhishek and the rest of you all. So, do you want to take a break now? Yes. Pushpa, all of these are PYQs. What is Friday, Pyarvani? Okay, Rohit, no problem. Take care. Do you want to take a break? Okay. It's 1 o'clock. Let's continue. Or do you want to take a break? Let me know. If you want a break, type A. If you don't want a break, type B. Pushpa has joined late. You want me to check yours? But don't worry about the rank and all of that if you've joined late. Let's see the majority and then decide whether or not we should take a break. All right, Vani, take care. Let's see. Okay, 62% don't need a break. Let's continue. Again, the majority does not need a break. Are you all not hungry? The next chapter that we have is Cell, the unit of life. In Cell, the unit of life, nothing is deleted. Why are you going to cry, Aditya? We'll take a break at 2 o'clock. How does that sound? Just give me 1 minute. Uh, just give me 1 minute. I'm coming. All right. So in this chapter, you learn about the different types of cells. I'm right here, Anvay. Prokaryotic cells, eukaryotic cells, the different types of organelles and structures found within cells. Mostly, you will get around 2 to 4 questions from this chapter. You can expect around 2 to 4 questions from this chapter. Either will ask you the structure of a cell organelle, or they will give you the function of the organelle and ask you for which organelle's function it is, or they will name the organelle and ask you for the function. So you basically get questions like that. Let's get started and start solving without wasting any more time. This is based on the concept of cell theory. The concept of omnis cellula acellula regarding cell division was first proposed by, this is a question from 2019, was it Aristotle, Rudolf Virchow, Theodor Schwann or Schleiden, German guy, I don't know how it's pronounced, Schleiden. Yes, as many of you are answering, option B is the correct answer. What does that mean? Omnis cellula acellula means what? If a new cell has to arise, it can only arise from pre-existing cells. New cells cannot form just out of the blue like that. You can't do some chumantar magic and make a new cell arise. If a new cell has to form, it has to form from pre-existing cells, and already existing cell has to undergo cell division, only then new cells will form, right? So that was proposed by Rudolf Virchow. 94% of you all have answered correctly. Jyoti, Anjali and Mona are in the top 3 spots. So basically the cell theory contribution, there are 3 scientists, Matthias Schleiden, Theodor Schwann and Rudolf Virchow. Matthias Schleiden was a botanist. He said that in plants are basically composed of many different types of cells, and those different cells form different types of tissues in plants. Theodor Schwann was a zoologist. Both of them worked around the same time, 1838-1839. He observed both plant cells and animal cells, and he noted that in the animal cells there is an outer plasma membrane, there is no cell wall, but in plant cells there is a cell wall. And then he also said that in the bodies of plants and animals, they basically composed of cells and its products. Rudolf Virchow kind of added another point to the cell theory by saying that if a new cell has to arise, it can only arise from pre-existing cells, right? So the correct answer for the previous question, like I mentioned, was option B. Now let's talk about our next question, which is asked in 2013. Pigment containing membranous extensions in some cyanobacteria are. I want you to remember that cyanobacteria are monerans. In cyanobacteria, which are prokaryotes, you do not find membrane-bound organelles. Cyanobacteria are photosynthetic. If photosynthesis has to happen in higher plants, it happens in chloroplast, because in chloroplast is where chlorophyll pigment is present. But these cyanobacteria, they are capable of performing photosynthesis, but they do not possess any membrane-bound organelles like the chloroplast. Then where are the pigments located? The pigments are located in extensions of the plasma membrane, which we call chromatophores. So the correct answer to this question is option B. Huh? What went wrong? Understood? Yeah, they are photosynthetic, autotrophs, unicellular, prokaryotic, correct. I don't know, something went wrong with the poll. It did not show anything. Let's take a look at our next question, which is from 2020 NEET. Which of the following statements about inclusion bodies is incorrect? Inclusion bodies are found in prokaryotes. Are they bound by any membrane? No, they are not. In prokaryotes, you do not find any membrane-bound structures. They are involved in the ingestion of food particles. They are not bound by any membrane is a correct statement. These are involved in ingestion of food particles. They are not involved. They lie freely in the cytoplasm. That is correct. These represent reserve material in the cytoplasm. That is also correct. So, correct answer here is option B. 57% have answered correctly. Sameer Raj, Anjali and Famida are in the top 3 spots. Here is our next question. Which of the following components provides sticky character to the bacterial cell? Which of the following provides sticky character to the bacterial cell? In the bacterial cell envelope, you have 3 different layers. You have a glycocalyx. Outside, inside that you have the cell wall, which is made up of peptidoglycan. Inside that you have the cell membrane, right? So this glycocalyx is not present in all bacteria. It is present only in some bacteria. This is what will provide it with a sticky characteristic. If you remember the experiment that we study about in molecular basis of inheritance, Griffith's experiment, Streptococcus pneumoniae, S strain and R strain and all of that. S strain is because of the presence of a capsule, which is basically the glycocalyx itself. So the correct answer here is option C. If the glycocalyx is present as a loose layer, you call it like the slime sheath. If it's a tough layer, you call it the capsule. Okay, so the correct answer here is option C. 77% have answered correctly. Sameer, Raj, Mona and Pushpa are in the top 3 spots. Here is our next question. Select the wrong statement. Select the wrong statement. Bacterial cell wall is made up of peptidoglycan. Is it? Is bacterial cell wall made up of peptidoglycan? Absolutely yes. Pili and fimbriae are mainly involved in motility of bacterial cells. Not at all. For motility, they will have flagella. Pili and fimbriae. Fimbriae helps in basically attaching the bacteria to colonies and substratum. Attaching. Pili, sometimes it helps in conjugation, a type of reproduction, sexual reproduction in bacteria. So this is incorrect. Cyanobacteria lack flagellated cells. That is true. Mycoplasma is wall-less microorganism. That is also true. So what is incorrect or wrong here is option B. So the correct answer here is option B. 85% of you have answered correctly. Preeti, Tejaswini, Sameer, Raj in the top 3 spots. Here we have our next question. Which of the following structures is not found in a prokaryotic cell? Do they have mesosomes? Do they have a plasma membrane? Is there a nuclear envelope? Do they have ribosomes? They are called prokaryotic because they have a primitive type of nucleus. A primitive type of nucleus is not covered by a nuclear membrane. So they do not contain a nuclear envelope. Mesosome, they will have their membranous extensions of the plasma membrane itself. All living organisms will have plasma membrane. Ribosomes are also present. So what is not present is your nuclear membrane or the nuclear envelope. So the correct answer is option C. 76% have answered correctly. Pushpa, Sameer, Raj and Tejaswini are in the top 3 spots. Here, let's take a look at our next question. The structures that help some bacteria to attach to rocks and or host tissues are mesosomes, holdfast, rhizoids, fimbriae. Mesosomes for sure are found in bacteria, but their function is not attachment. Holdfast is something that you find in brown algae, holdfast, type and frond. That helps in attachment in brown algae, not in bacteria. Rhizoids help in attachment, in bryophytes. And in bacteria, what helps in attachment is fimbriae. So the correct answer to this question is option D. 73% of you all have answered correctly. Tejaswini, Pushpa and Mona are in the top 3 spots. Let's take a look at our next question now. What do you mean this lecture is from where? I don't understand your question, Pushpa. Which structures perform the function of mitochondria in bacteria? Which structures perform the function of mitochondria in bacteria? Can the nucleoid perform the function of bacteria? No. Do the ribosomes function? No. No. Yes. What are these mesosomes? If this is the cell wall in bacteria, inside that you have a cell membrane. There will be extensions of the cell membrane like that, which we call mesosomes. How do these mesosomes? How do these mesosomes function like mitochondria? What is the function of mitochondria? To produce ATP. How does it produce ATP? With the help of ATP synthase. Where is this ATP synthase present? It is present in the inner mitochondrial membrane. The inner mitochondrial membrane structure is very similar to the structure of your plasma membrane. Just like you have an ETC present in the inner mitochondrial membrane, you will have enzymes like ATP synthase present in these regions, in mesosomes, and that is where ATP gets synthesized. So that's why they perform functions similar to mitochondria. Option D is your correct answer here. Hopefully everyone's understood. 12 students, that is 80%, have answered correctly. Pushpa, Anvay and Aditya are in the top 3. Next question from 2023. Which of the following functions is carried out by cytoskeleton in a cell? What does cytoskeleton help in? Yeah. Just give me a minute. I'll drink some water and come back. Just 1 minute. So yeah, it helps in motility. Correct answer is option A. 90% have answered correctly. Akash, Aditya and Sharat are in the top 3 spots. If you are going to keep socializing, chatting, making jokes here in the comment section, I'll have to have you blocked. Please do not distract the others who are here to study. If you have to have fun, then you can find some other platform for that. This is not the place. Let's take a look at our next question here. Which of the following are not considered as a part of endomembrane system? In eukaryotes, there are some membrane-bound organelles whose functions are related, interrelated, coordinated. So, all of them are grouped together as the endomembrane system. Among the given organelles, which is not a part of the endomembrane system, mitochondria is not, ER is, chloroplast is not, Golgi is, peroxisomes are not. So, B and D is the correct answer, which is option C. This is asked in 2023, last year. Only 10% have answered correctly. That is just 2 students, Aditya and Muthukrishnan. Now let's move on. How many different proteins does the ribosome consist of? Actually, this information is there in molecular basis of inheritance chapter. It's not there in cell, the unit of life. Ribosomes are basically composed of a type of RNA called ribosomal RNA and proteins. There are about 80 different types of proteins present in ribosome according to the data in NCERT. So the correct answer here is option C. 73% have answered correctly. Sameer, Raj, Shri and Mona are in the top 3. Here is our next question. This is based on the different types of chromosomes. This is a question from 2022. Very simple question. Metacentric chromosome will have centromere in the middle and form 2 equal arms, right? So, this is 3. Acrocentric chromosomes will have one very large and one very small arm. Extremely short and one extremely long arm. This is 1. Submetacentric is slightly away from the center. One shorter, one longer arm. This is 4. Telocentric, centromere is present at the tip, at the terminal end. So this is 2. Correct answer here is A3B1. C4D2. I've been noticing every time we are solving match the following questions in today's class, in most cases option A itself is the correct answer. How many of you noticed? 95% have answered correctly. Akash, Preeti and Lahari are in the top 3 spots. Here is our next question. Which of the following statements with respect to endoplasmic reticulum is incorrect? Which question? Okay, I'll check after this question. Which of the following statements with respect to endoplasmic reticulum is incorrect? Rough endoplasmic reticulum has ribosomes attached to it. Yes. Smooth endoplasmic reticulum is devoid of ribosomes. Yes. In prokaryotes, only rough endoplasmic reticulum is present. No. In prokaryotes, no endoplasmic reticulum is present. So, they don't have any membrane-bound organelles, so that is incorrect. Smooth endoplasmic reticulum is a site for lipid synthesis. That is correct. What is incorrect here is option C. So, option C is the correct answer. 41% of you have answered correctly. Anurag, Pushpa and Bharat have answered. They are in the top 3. Anvay says that I have not answered one question properly, which is that, let me check. B and D. Yeah, what's wrong here? Mitochondria, chloroplast, peroxisomes, they're not part of the endomembrane system. Not a part. Right. So it is A, C and E, which was option D. You're right. Sorry. Here is our next question. Which of the following is an incorrect statement? It's a statement based question from 2021. Nuclear pores act as passages for proteins and RNA molecules in both directions between the nucleus and the cytoplasm. That is correct. That is correct. Mature sieve tube elements possess a conspicuous nucleus and usual cytoplasmic organelles. No, that is incorrect. They will not have a nucleus. They are enucleated cells similar to our RBCs. This is the incorrect statement. Microbodies are present in both plants and animal cells. That is true. They will have certain enzymes. The perinuclear space forms a barrier between the materials present inside the nucleus and that of the cytoplasm. This is also true. Perinuclear space is basically the space that is present between the two nuclear membranes, right? So, correct answer here is option B. That is the incorrect statement. 78% of you have answered correctly. Akash, Preeti, Anvay in the top 3. Here is the next question. Cristae. What are cristae? Where do you find cristae? Cristae are found in mitochondria. Inner membrane of the mitochondria forms infoldings and form cristae. So P can be matched with 3. Thylakoids. Thylakoids are present in plastids like chloroplast. They are flattened membranous sacs that are found in the stroma of plastids like chloroplast. So Q is matched with 4. Centromere is the primary narrow region of the constriction in the chromosome. This is matched with 1. Cisternae are, what else do we have left? Yeah, disc-shaped sacs present in the Golgi apparatus. Correct. So correct answer here is P3, Q4, R1, S2, which is option D. For a change, we have 84% have answered correctly. Aditya, Sameer, Raj and Pushpa in the top 3. Here is our next question. When the centromere is situated in the middle of two equal arms of chromosomes, the chromosome is referred as. If it is in the middle, what do we call it? If it is in the middle, we call it metacentric. If it is slightly away from the center, we call it submetacentric. If it has like one extremely long, one extremely short arm, we call it acrocentric. If the centromere is present at the tip, we call it telocentric. Middle of two equal arms, then we call it metacentric. So option B is the correct answer. 80% of you have answered correctly. Sameer, Raj, Mona and Aditya in the top 3. Next question. The organelles that are included in the endomembrane system are. This is a question from 2021. Which of these organelles are included in the endomembrane system? Golgi is, endoplasmic reticulum is, mitochondria is, and lysosomes are. ER is no mito, no ribo, lyso. ER, Golgi, lyso, vacuoles. Golgi, mito, ribosomes, lysosomes. So correct answer here is option C. 81% have answered correctly. Mona, Anvay and Anurag are in the top 3. Let's move on and take a look at our next question. Which is the important site of formation of glycoproteins and glycolipids in eukaryotic cells? Does it happen in endoplasmic reticulum or peroxisomes or Golgi bodies or polysomes? Formation of glycoproteins and glycolipids, what basically happens is sugars are added to proteins and to lipids. The process is called glycosylation. It happens in our Golgi bodies. So the correct answer here is option C. 80% of you have answered correctly. Anvay, Pushpa, Anurag, top 3. This is a question from 2020. Now let's take a look at our next question, which is statement based. Which of the following statements is not correct? Lysosomes are formed by the process of packaging in the endoplasmic reticulum. Are they formed in the endoplasmic reticulum? No, they are formed in the Golgi apparatus. We already found our correct answer, but let's just go through the other options as well. Lysozymes have numerous hydrolytic enzymes. Yes, there are many of them that are capable of breaking down our proteins, carbohydrates, and all of that. The hydrolytic enzymes of lysosomes are active under acidic pH. That is also true. Lysosomes are membrane-bound structures. That is also true. They are bound by a single membrane. So what is not correct here is option A. So the correct answer here, let's see how many of you answered. 57% have answered. Preeti, Anvay and Pushpa in the top 3. Here is our next question. Which of the following statements regarding mitochondria is incorrect? Mitochondrial matrix contains single circular DNA molecule and ribosomes. Yes, it does. Outer membrane is permeable to monomers of carbohydrates, fats and proteins. Yes. Enzymes of electron transport are embedded in the outer inner membrane is convoluted with infoldings. We call that cristae, right? Enzymes of ETS are embedded not in the outer membrane, they are embedded in the inner membrane. That is what is incorrect. So the correct answer to this question is option C. 33% have answered correctly. Akash, Anvay and Aditya, all names starting with A. Here is our next question. The shorter and longer arms of submetacentric chromosomes are referred to as M arm and N arm respectively, S and L, P and Q, Q and P. Shorter one is P, longer one is Q. Actually, this is not mentioned in your NCERT textbook. Petite, P for petite. Petite means small, I think in French. I don't know if that's how it's pronounced. So, shorter one is P. Longer one is Q. So correct answer here is option C, P arm and Q arm respectively. Preeti, Anurag and Pushpa in the top 3. Let's take a look at our next question. Which of the following pairs of organelles does not contain DNA? Which of the following pairs of organelles does not contain DNA? Nuclear envelope, within that DNA is there. In mitochondria also there's DNA. In mitochondria there's DNA, in lysosome there's no DNA. In chloroplast there's DNA, in vacuoles there's no DNA. In lysosomes there's no DNA, vacuoles also there's no DNA. So correct answer is option D. %70 have answered correctly. Aditya, Mona, Anvay in the top 3. Let's move on and take a look at our next question. It is a match the following type of question. Under column 1, different organelles are listed. Under column 2, their functions are given. You have to match it correctly. Golgi apparatus is involved in the formation of glycoproteins and glycolipids. Lysosomes help in digesting biomolecules. Vacuoles trap waste and excretory products. Ribosomes are involved in the synthesis of proteins. So correct answer here is A3, B4, C2, D1, which is option A. 59% have answered correctly. Anurag, Anvay, Jyoti in the top 3. Which of the following is true for nucleolus? Larger nucleoli are present in dividing cells. In dividing cells, nucleolus disappears, right? So this is incorrect. It is a membrane-bound structure. No, it is not bound by any membrane. It takes part in spindle formation. It doesn't. It forms by spindle formation happens with help of microtubules. And it is a site for active ribosomal RNA synthesis, rRNA synthesis. This is correct. So the correct answer to this question is option D. 18% only have answered correctly. Pushpa, Bharat and Sharat are in the top 3. Okay, Anjali, no problem. All the best. Here is our next question. The Golgi complex participates. The Golgi complex participates in fatty acid breakdown, formation of secretory vesicles, respiration in bacteria, activation of amino acid. It helps in formation of secretory vesicles, right? So the correct answer to this question here is option B. 71% of you all have answered correctly. Anvay, Anurag and Jyoti in the top 3. Jyoti has been answering most questions correctly. Congrats, Jyoti. Next question. Which of the following events does not occur in rough endoplasmic reticulum? Protein folding, protein glycosylation, cleavage of signal peptide, phospholipid synthesis. On rough endoplasmic reticulum, ribosomes will be present. So these ribosomes, after producing proteins, can transport them into the lumen of the ER, where the protein can undergo folding. Sometimes it adds sugar to the protein also and perform protein glycosylation. Signal peptide also is cleaved. Okay, so basically at the end terminal of your polypeptide, there will be a short sequence which will be cleaved in the endoplasmic reticulum, but phospholipid synthesis does not happen here. So correct answer here is option D. 50% have answered correctly. Sharath, Mona and Sweetie in the top 3. Here is our next question. Which of the following cell organelles is responsible for extracting energy from carbohydrates to form ATP? Which of the following cell organelles is responsible for extracting energy from carbohydrates to form ATP? It is our mitochondria, which is called the powerhouse of the cell. Yes. So option C is the correct answer here. 84% have answered correctly. Akash, Pushpa and Anvay in the top 3. Next question. From NEET 2016 phase 2. Select the mismatch. Gas vacuoles are found in green bacteria. Large central vacuoles are found in animal cells. Protists are eukaryotes. Methanogens are prokaryotes. So the correct answer here is option B. In animal cells, you do not find a large central vacuole. You find that in plant cells. That is the mismatch. Correct answer is option B. 59% have answered correctly. Jyoti, Pushpa and Mona in the top 3 spots. Here is our next question. A cell organelle containing hydrolytic enzymes. What are these hydrolytic enzymes? Hydrolytic enzymes make use of water to break bonds, right? For lysis, they make use of water. Such enzymes are found in the suicide bags of the cell, which are lysosomes. So the correct answer to this question is option A. 76% of you all have answered correctly. Jyoti, Anvay and Sharath in the top 3. Next question. Mitochondria and chloroplast are they semi-autonomous organelles? Yes, they are. formed by the division of pre-existing organelles, and they contain DNA but lack protein synthesizing machinery. No, they contain DNA and they also contain protein synthesizing machinery. That makes this statement incorrect. Of the given two statements, A is correct, B is incorrect. So option A is the correct answer. 40% of you all have answered correctly. Ayushman, Ansh and Aditya in the top 3. Alright, now let's move on and take a look at our next question. Microtubules are the constituents of. Microtubules are the constituents of. Centrioles, spindle fibers, chromatin, no. Centrosome, fusosome, centrioles, cilia, flagella, peroxisome, fibers, centriole, cilia. Option D is the correct answer. 60% have answered correctly. Ansh, Naga and Jyoti in the top 3. Welcome Naga, long time. How are you doing? Why did you join so late? Anva is confused between D and C. Next question. Which one of the following cell organelles is enclosed by a single membrane? Nuclei, mitochondria, chloroplast, all of these are bound by two membranes. Lysosomes have a single membrane. So the correct answer here is option A. 80% have answered correctly. Jyoti, Peace and Anvay in the top 3. Rohit is also back. Let's move on to our next question for the day. Water-soluble pigments found in plant cell vacuoles are, okay, this is out of syllabus. This was asked in NEET 2016 phase 1. Generally, pigments are found inside of plastids, right? But here they are asking which is that pigment which is soluble in water and it's present in vacuoles. It is anthocyanins. So correct answer here is option B. 28% of you have answered correctly. Pushpa, Cooking and Simran in the top 3. Next question, very similar question we've already solved before. This is from 2015 NEET. Match the columns and identify the correct option. Under column 1, you have different parts of different organelles, and under column 2, you have their description. You have to match it correctly. Where do we find thylakoids? We find it in chloroplast, specifically in the stroma. So they are flat membranous sacs that you find in the stroma. So A can be matched with 3. Cristae are the infoldings of the inner membrane of mitochondria. So B is matched with 4. Cisternae are disc-shaped sacs that you find in Golgi apparatus. That is 1. Chromatin is condensed structure of DNA. So that is 2. So correct answer here is A3, B4, A3, B4, C1, D2, which is option D. 19% have answered correctly. Sweeties, Mona and Jyoti in the top 3. What was the doubt here? Why did so many of you answer it incorrectly? Okay. So be careful. Okay. Don't do jaldi bazi then. Leaderboard after this chapter we'll do. We have like a few more questions left, I guess. Yeah, like 3 more questions left. After that we'll check the leaderboard. DNA is not present in In the nucleus it's there, in the mitochondria it's there, in the chloroplast it's there, in ribosome it's not there. In ribosome you have RNA, you don't have DNA. So the correct answer here is very simple. It is option D. %79 have answered correctly. Jyoti, Mona, Pushpa in the top 3. Next question. Which of the following types of plastids does not contain stored food material? Stored food is not present in which of the following? In chromoplast, do you have stored food? No, you don't. In elaioplast, you have stored food. Fats or lipids are stored here. In aleuroplast, proteins are stored. In amyloplast, starch or carbohydrates are stored. So all of these are types of leucoplasts whose function itself is to store. Chromoplasts are plastids that contain pigments other than chlorophyll, the green pigment. So correct answer here is option A. 80% have answered correctly. Anurag, Pushpa and Jyoti are in the top 3. Let's take a look at our next question, which is from 2012. Which one of the following does not differ in E. coli and Chlamydomonas? First of all, we need to understand what is the difference between E. coli and Chlamydomonas. E. coli is a prokaryotic cell. Chlamydomonas is eukaryotic. Are the ribosomes different? Yes, they are different. In E. coli, you have 70S ribosomes. In Chlamydomonas, you have 80S ribosome. Chromosomal organization or the DNA organization also is different. Cell wall is also different. It's peptidoglycan here. In Chlamydomonas, it is cellulose. Cell membrane is the same, right? The correct answer is option D. 84% of you will have answered correctly. Anurag, Pushpa and Anvay in the top 3. Okay, so with that we have finished the important PYQs from the chapter Cell the Unit of Life. Hopefully you were able to revise your concepts well. Let's take a look at the leaderboard. Ram is still on top of the list. He has answered 118 out of 136 questions correctly. Then we have Suresh and then we have Rohit, who is in the 3rd spot right now. Then we have Anjali, Tejaswini, Mona, Jyoti, Soni, Sadhana, Vishal, Achal, Lisa, PMS, Only Studies, Boring Games or Aditya, Tanmay, Sweetie, Ansh, Pushpa, Mayuri in the top 20. 21 we have Samir Raj and then we have Bharat in 22. Then we have Easy Math 23, Shirisha, Shri, Anva in 26, Anurag 27. Alright, so yeah. Pushpa, I just showed your name, no? Now not 2 chapters, 4 more chapters left. Cell the Unit of Life is over. Cell cycle and cell division. And then we have photosynthesis in higher plants. Then we have respiration in plants. Then we have plant growth and development. Okay. So what is the time now? The time is 1:50. Let's take like a little break. How many of you want a break? Pushpa, I showed you, no? Did you not see? What time is it? 2 o'clock. Let's finish it off. If anyone's hungry, just go grab like some food from the kitchen, come and sit in your study room and you can eat while you watch. It's not like you have to talk like me, no? Yeah, let's finish it off in one go. Thank you so much, Rohit. It means so much. It feels like all the effort we put in is worth it when we hear such things. Hopefully, you'll get very good marks also. Okay, so in cell cycle and cell division, we learn about mitosis, meiosis, the process, and what is the significance of mitosis and meiosis, what is the cell cycle and all of that. Okay, thanks Vaishnavi. Hi Deepu, good afternoon. Cooking Fantasies is becoming impatient. Okay, let's start. Okay, so this is a small chapter but carries a lot of weightage. 4 to 5 questions can be expected from this chapter. Okay, so here is our first question, which is a match the following type of question. This was asked last year in 2023. What happens in the different phases of the the cell cycle? What happens during M phase? Equational division takes place. What happens during G2 phase? Proteins are synthesized. What happens during quiescent stage, which is your G0 phase? It is inactive with respect to cell division. G1 phase is the interval between mitosis and the initiation of DNA replication. So this is 3. Initiation of DNA replication happens in your S phase, right? So the correct answer here is A4, B1, C2 and D3, which is option A. Okay, 64% of you have answered correctly. Cooking Fantasies, Mona and Venkateshan have answered correctly and are in the top 3 spots. Here is our next question. DNA replication in bacteria occurs in. Why not? DNA replication in bacteria occurs in. Within the nucleolus? No. Prior to fission? Just before transcription? No. During S phase? No. In bacteria, mitosis will not take place, amitosis will take place. In amitosis, there is no chromosome formation and all of that. So, it undergoes binary fission. So, just before it undergoes binary fission, replication of DNA will take place. So, option B is your correct answer. 11% of you all have answered correctly. Jyoti and Pushpa, just the 2 of you. Next question. This again is a question from 2023. Deepu is asking how you should join the poll. In order to join the poll and participate in this quiz, you have to first enroll in something called Unity Portal. The link to that is pinned at the top of the chat box. Just whichever chat box you're typing in right now, check at the top, you'll have a link. Click on that link. It should not take you more than 30 seconds. It'll just ask you for some credentials. Once you enter that, you will enroll in Unity. After that, whenever the questions are displaced, whatever you whatever you think is the correct answer, type just that option in the chat box. The Unity portal will pick it up and your name will be displayed in the poll, okay, in the leaderboard. Alright. If you don't enroll in Unity and you type your options here, correct answers here, the poll will not be able to pick it up. It will not reflect here. Okay. Among eukaryotes, replication of DNA takes place in which phase? Very simple question. It happens in S phase. S is for synthesis here. What gets synthesized? DNA gets synthesized, and also if it's an animal cell, then the duplication of centrioles also will happen. So the correct answer here is option D. 89% of you all have answered correctly. Jai Shri Krishna, Mona and Anvai in the top 3. Next question. Given below are 2 statements. It's a statement based question. Statement 1. During, okay, so this should be G0 phase, I guess, GS phase, there is a typo. During G0 phase of the cell cycle, the cell is metabolically inactive. Statement 2. The centrosome undergoes duplication during S phase of interphase. Over here in the G0 phase, we call it quiescent stage, which is an inactive stage. We say it is inactive because it's not actively dividing, but that does not mean that the cell is metabolically inactive. If it's metabolically inactive, it's as good as dead, right? So over here in the G0 phase, the cell will be metabolically active. That makes the statement incorrect. Statement 2, centrosome undergoes duplication during S phase. That is true. So statement 1 is incorrect, but statement 2 is correct. Option B is the correct answer. 79% of you will have answered correctly. Naga, Vaishnavi and Anvay in the top 3. Next question. Again, we have a match the following type of question. This is from 2021. Okay, all the best, Pushpa. S phase, replication of DNA. G2 phase, hey, we already solved this, no? Is this like a repetition? Quiescent stage. I think this is repeated. Four, 1, 2, 3. I think the slide itself is repeated. Option D is the correct answer. 54% have answered correctly. Naga, Mona, Vaishnavi in the top 3. Let's take a look at our next question. The fruit fly has 8 chromosomes, that is the diploid number in each cell. During interphase of mitosis, if the number of chromosomes at G1 phase is 8, what would be the number of chromosomes after S phase? So you have in your cell cycle, you have interphase and then you have your M phase. In interphase again, you have G1, you have S, you have G2. In G1, they are saying that the number of chromosomes is 8. In S phase, there is DNA replication. They are asking after S phase, what would be the number of chromosomes? The number of chromosomes would still be 8. The DNA content would have doubled, but the number of chromosomes will not change. Over here, in the G1 phase, if each chromosome has 1 chromatid, after replication, each chromosome will have 2 chromatids. This is also 1 chromosome, this is also 1 chromosome. The difference is, before S phase, it will have only 1 chromatid, but after S phase, each chromosome will have 2 chromatids. Over here, you call this the 2C condition, you call this the 4C condition. Whenever we count the number of chromosomes, we don't look at the number of chromatids, we always look at the number of centromeres. If there's 1 here, that's 1 chromosome, there's 1 here, that is 1 chromosome. Okay, the DNA content will increase, but the number of chromosomes will not increase. So the correct answer to this question is option B. Most students get confused in this concept. Hopefully you've understood. 52% Naga, Jayashree Krishna and Sharath in the top 3. Next question. Very simple direct question. The centriole undergoes duplication during G2 phase, S phase, prophase, metaphase. Forming an extra copy of the centrosome in animal cells or the centriole happens in your S phase while the DNA is replicating inside the nucleus. Centriole will undergo duplication in the cytoplasm. So the correct answer here is option B. 72% of you all have answered correctly. Mona, Nazrit and Bhairavi Venkateshan have answered, they're in the top 3. This is a question from 2021. Let's take a look at our next question here. Identify the correct statement with regard to G1 phase of interphase. Which is correct? We have to identify. This is a question from 2020. DNA synthesis or replication takes place. DNA synthesis or replication takes place in G1 phase? It doesn't. Reorganization of all cell components takes place? No. Cell is metabolically active. It grows but does not replicate its DNA. This is correct. Nuclear division takes place. Nuclear division does not take place here. It takes place in the M phase. So what is correct? Option C is correct. 94% of you all have answered correctly. Jyoti, Naga and Mona in the top 3. Sweetie is calling out for Sebastian, who is not even here. Only Indian number is available. Deepu, I don't know which number is yours. Alright, let's move on and take a look at our next question, which is again from the year 2020. Some dividing cells exit the cell cycle and enter a vegetative inactive stage. This is called quiescent stage. It is represented by G0. This process occurs at the end of. Actually, it does not occur at the end of one phase. It occurs somewhere in between. M phase, G1 phase, S phase, G2 phase. It happens in the G1 phase. What basically happens during G1 phase is the cell would have just then divided and it will be like one baby kutti cell. This is G1 phase, this is S phase, this is G2 phase, this is your M phase. So this is this baby cell. Throughout the course of G1 phase, it will increase in size, and once it increases so much that the single nucleus cannot handle all of the activities in the cytoplasm, it will decide, okay, I have to divide, and before it undergoes division, it will go to the S phase and make an extra copy of its genetic material. But what happens in cells that don't constantly undergo cell division is it will grow to a certain extent. It will not increase too much in size, not so much that the nuclear cytoplasmic ratio gets disturbed. It will increase considerably in size and it will exit the G1 phase and it will be in a inactive stage. Inactive meaning it's not actively undergoing cell division, but it's metabolically active. That is your G0 phase. So G0 phase exits the cell cycle from the G1 phase. So option B is the correct answer. Alright, everyone's answered correctly. Mona and Naga and Pranav in the top 3. Here is our next question. Cells in G0 phase terminate the cell cycle, exit the cell cycle, enter the cell cycle, suspend the cell cycle. They exit the cell cycle. Correct answer here is option B. 74% of you all have answered correctly. Mona, Pranav and Nazrit in the top 3. Let's take a look at our next question. The correct sequence of phases in the cell cycle is? What is the correct sequence? G1 first, then S, then G2, then M. Right? The correct answer here is option A. 74% have answered correctly. Jyoti, Mona and Naga in the top 3. Let's take a look at our next question from NEET 2016 phase 2. During cell growth, DNA synthesis takes place, it should be in, takes place in S phase, G1 phase, G2 phase, M phase. Very simple question. We've already answered similar questions, right? So DNA synthesis takes place in your S phase itself. So the correct answer to this question is option A. Mona, Jyoti and Naga are in the top 3. Let's take a look at our next question from 2014. During which phase of cell cycle amount of DNA in a cell remains at 4C level if the initial amount is denoted as 2C? During which phase of cell cycle amount of DNA remains at 4C level if the initial amount is denoted as 2C? Option C is the correct answer, because in G1 it is 2C, in S phase initially starting it is 2C, after DNA replication towards the end of S phase it will be 4C, so it does not remain 4C throughout. In G2 it remains 4C throughout. In M, at the beginning of division, before the nuclear division happens, before the splitting of centromere happens, you have 4C. Towards the end of M phase you will have 2C, so it does not remain 4C. It's only in the G2 phase that it remains 4C throughout. So the correct answer to this question is option C. 21% of you all have answered correctly. Anurag, Deepika and Rohit in the top 3. Any confusions here? Okay, Aditya. Here is our next question. In the S phase of cell cycle, amount of DNA doubles in each cell, amount of DNA remains the same in each cell, chromosome number is increased, amount of DNA is reduced to half in each cell. Amount of DNA doubles, but the chromosome number will not increase. So the correct answer is option A. Are you keeping a track, or are you noticing how so many questions are based on S phase itself? 70% of you all have answered correctly. Pranav, Mona and Sweetie in the top 3. Next question. Which one of the following never occurs during mitotic cell division? Which one does not occur during mitotic cell division? This is from 2022. Spindle fibers attach to kinetochores of chromosomes. Does it happen during mitosis? Yes, it does. Movement of centrioles towards opposite poles. If it's an animal cell, that also will happen. Pairing of homologous chromosomes does not happen during mitosis. That is an event that happens during meiosis. Coiling and condensation of the chromatids, this also happens. What doesn't happen is option C. 81% of you all have answered correctly. Anurag, Jyoti and Sweetie in the top 3. Next question. Select the incorrect statement with reference to mitosis. All the chromosomes lie at the equator at metaphase. Yes, it does. Spindle fibers attach to centromere of chromosomes. It attaches to the kinetochore. Chromosomes decondense at telophase. Splitting of centromere happens at anaphase. That is correct. So option B is incorrect here. 57% have answered correctly. Naga, Sheikh Khalil and Mona in the top 3. Next question from 2017. This is based on the events of meiosis. Which of the following options gives the correct sequence of events during mitosis? During mitosis. Whichever options have crossing over, you can eliminate that because crossing over does not happen in mitosis. You have option A and option C. Condensation, nuclear membrane disassembly, arrangement at equator, centromere division, segregation, telophase. Okay, A is the correct answer. %75% have answered correctly. Naga, Mona and Sweetie, top 3. Next question. Spindle fibers attach onto centromere of the chromosome, kinetosome of the chromosome, telomere of the chromosome, kinetochore of the chromosome. Spindle fibers attach onto your kinetochores. These are basically paired disc-like structures that are present on either sides of your centromere. So correct answer here is option D. Mona, Naga and Sheikh, top 3 spots. Next question. Which of the following is not a characteristic feature during mitosis in somatic cells? Which of the following is not a characteristic feature during mitosis in somatic cells? Chromosome movement happens during anaphase. Spindle fibers form. Nucleolus disappears during prophase. Synapses occurs in meiosis, not during mitosis. So the correct answer here is option B. 72% have answered correctly. Sweetie, Naga and Mona in the top 3. Next question. The process of appearance of recombination nodules occurs at which substage of prophase 1 in meiosis? The process of appearance of recombination nodules occurs at which substage of prophase 1? The process of appearance of recombination nodules occur at which stage? In leptotene, condensation begins. Zygotene, synapses. Pachytene. Option D is the correct answer. Crossing over happens in pachytene. At the sites of crossover, recombination nodules will form. Why Sagar Mandal saying RCB? Option D is the correct answer here. Mona, Nahid Sheikh and Jayashri Krishna in the top 3. Next question. Yeah, I just had some water. Thanks, Rohit. Which of the following stages of meiosis involves division of the centromere? In meiosis, where does centromere division happen? Anaphase 2, telophase, metaphase 1, metaphase 2. It happens during anaphase 2. Centromere splitting does not happen during anaphase 1 in meiosis. In anaphase 1, homologous chromosomes separate, but in anaphase 2, the centromere will split and the chromatids will separate. So the correct answer to this question is option A. 76% of you all have answered correctly. Naga, Jayashree Krishna and Deepika in the top 3. This is a question that was asked in 2023. Let's take a look at another question that was asked in NEET 2023. Okay. Sagar has the trick for you all to remember. Sony, can you try enrolling in Unity one more time, Sony? Select the correct statements. Tetrad formation is not seen during leptotene. During anaphase, the centromere split and the chromatids are separate. Yes. Terminalization takes place during pachytene. No, it happens during diakinesis. Nucleolus, Golgi complex and ER are reformed during telophase. Yes. Crossing over takes place between sister chromatids, non-sister chromatids of homologous chromosomes. So this is also incorrect. Choose the correct statement andre B and D only. So option D is the correct answer. 47% of you all have answered correctly. Naga, Sheikh and Famida in the top 3. Next question. The appearance of recombination nodules on homologous chromosomes during meiosis characterizes. Deepika, please enroll in the Unity portal if you have not already enrolled. At the top of the chat box, there is a pinned comment. There you have a link called Unity. You have to click on that link and enter your credentials. It will take like 20 seconds to do that. Once you have enrolled there, it will start reflecting in the leaderboard here. The appearance of recombination nodules on homologous chromosomes during meiosis characterizes synaptonemal complex, bivalent. Yeah, sites at which crossing over occurs, not terminalization. So the correct answer here is option C. 50% have answered correctly. Anurag, Naga and Jayashree Krishna in the top 3. Let's take a look at another question from 2022. Jayashree Krishna's name is Nishika. Regarding meiosis, which of these statements is incorrect? What is not correct? There are 2 stages in meiosis. Yes, there's meiosis 1 and meiosis 2. DNA replication occurs in S phase of meiosis 2. Not really. There is no S phase before meiosis 2. There's just one S phase before meiosis 1. Between meiosis 1 and meiosis 2, you just have interkinesis, a brief period where replication of DNA does not take place. So that is incorrect. Pairing of homologous chromosomes and recombination occurs in meiosis. Yes, 4 haploid cells are formed at the end of meiosis 2. Yes, that is also correct. Reductional division will happen if the parent cell is diploid. Then at the end, 4 haploid cells will form. So correct answer here is option B. 71% of you all have answered correctly. Naga, Suraj and Mona in the top 3 spots. Can you hear my son screaming? Where are you going, sweetie? Don't go. Which of the following stages of meiosis involves the division of centromere? Centromere division happens when? In meiosis, in anaphase 2. Like I already mentioned, in anaphase 1, only the homologous chromosomes will separate. The centromere will not split. In meiosis 2, anaphase 2, centromere splitting happens. Okay, one sweetie is gone, another sweetie is still there. Thank you. So the correct answer here is option D. 75% have answered correctly. Nazrit, Naga and Jayashree Krishna. You said your name was something. Jayashree Krishna's name is, I forgot. Sorry. Nishika. Okay. 75% have answered correctly, right? Yeah. ಕಿರ್ಚಬೇಡ ಅಂತ ಹೇಳು ಪವಿ ಅವನ್ಗೆ. Did you start answering already? I didn't start the poll yet. Which stage of meiotic prophase shows terminalization of chiasmata as its distinctive feature? It happens in diakinesis. So correct answer here is option D. Yeah, poll had not yet started, my bad, sorry. Anurag, I'm in Yakin 2025 batch, I'm there. I had not yet joined the organization before this Yakin was launched last year. So option D is the correct answer here. 93% have answered correctly. Sweetie, Deepika, Rohit in the top 3. Next question. Dissolution of the synaptonemal complex occurs during. Dissolution of the synaptonemal complex occurs during pachytene, zygotine, diplotene, leptotene. Dissolution happens when? During diplotene, except for the sites of crossover. In other places it will start separating. That will form X-shaped structures called chiasmata. Right? So the correct answer to this question here is option C. 53% have answered correctly. Nazrat, Naga and Bhairavi are in the top 3. Next question, again from 2020. Match the following with respect to meiosis. What are you saying, Rohit? Sagar Dvan nahi diya. What does that mean? Rohit. Zygotine synapsis, pachytene crossing over, diplotene chiasmata, diakinesis terminalization. 4, 3, 2, 1. Option B is the correct answer. 75% have answered correctly. Naga, Deepika, Mona in the top 3. Next question from 2019, Odisha NEET. Crossing over takes place between which chromatids and in which stage of the cell cycle? Crossing over happens in prophase 1. In every option, prophase 1 is given. Does it happen between sister chromatids or non-sister chromatids? Does it happen between homologous chromosomes or non-homologous chromosomes? It happens at prophase 1. It happens between homologous chromosomes. So these two options you can eliminate. It happens between non-sister chromatids of homologous chromosomes. Leptotene, zygotene, pachytene is the correct answer. Option B is the correct answer here. 76% have answered correctly. Naga, Deepika, Vaishnavi in the top 3. Next question. The stage during which separation of the paired homologous chromosomes begins is. Paired homologous chromosomes will start to separate. Dissolution of synaptonemal complex we solved in another question, right? When do the homologous chromosomes that have paired start to separate? In Pachytene, crossing over is already over. In Diplotene, they will start coming apart. So the correct answer to this question is option B, Diplotene. 41% have answered correctly. Naga, Nazareth and Mona in the top 3. Next question from 2016. Match the stages of meiosis in column 1 to the characteristic features in column 2 and select the correct option using the codes given below. See, very similar, repeatedly, same kind of questions have been asked over and over again. What happens during Pachytene? Crossing over will take place. What happens during metaphase 1? Chromosomes will align at the equatorial plate. Diakinesis, terminalization of chiasmata, pairing of homologous chromosomes or synapses happens in zygotene. So 3, 4, 2, 1 is the correct answer and it is option A. 67% have answered correctly. Preeti, Nishika, Sagar in the top 3. Next question again from 2016, phase 1. In meiosis, crossing over is initiated in which phase? Zygotene, Diplotene, Pachytene, Leptotene. Crossing over is initiated in which phase? Many of you are answering as Pachytene and Pachytene itself is the correct answer. So option C is right. 84% have answered correctly. Sagar, Anurag and Nishika in the top 3. Next question, again based on the same concept, very similar type of question from 2015. Arrange the following events of meiosis in the correct sequence. What happens first? Synapses, crossing over, terminalization of chiasmata, disappearance of %31 have answered correctly. Nishika, Nazareth and Naga. Next question. Again, it's another match the following question from 2015. Select the correct option. Under column 1, you have the different events. Under column 2, you have the different stages. Synapses aligns homologous chromosomes. When? In zygotene. When does synthesis of RNA and protein take place? In your G2 phase. Action of enzyme recombinase. There should have been Pachytene. Centromeres do not separate, but chromatids move towards opposite poles. Centromeres do not separate, but chromatids move towards opposite poles as anaphase 1. Action of enzyme recombinase should be Pachytene, no? 2, 3. There should have been a 5 here because in the option they've mentioned 5, but this person who typed this thing probably skipped that. So this 5 should be Pachytene, right? Then it makes sense. Then this would be 5, right? So 2, 3, 5, 4. 2, 3, 5, 4 is option D. Yeah, 40% have answered correctly. Vaishnavi, Naga, Nazareth in the top 3. Yeah, with that we have finished cell cycle and cell division. Suresh is on top of the list. Ram is second right now. So there was very close competition between the two of you. Then we have Rohit in the third spot. Then we have Mona and Anjali and Tejaswini and Jyoti, Soni, Dr. Soni. I think you were asking, your name is not seen. You can see your name now. Then we have Sweetie, Aditya, Sadhana, Vishal, Achal Singh, Lisa, PMS, Only Studies, Tanmay, Pushpa, Bharat, Ansh, Mayuri, Sameer Raj, Naga, Bhairavi, Fahmida, Nishika, another Sweetie, Anurag, and the rest of you all. So, yeah, I did show your name. Naga, did you not see? Bonus mark because of typing mistake. It's not like the question is wrong, it was typed incorrectly. All right. So let's move on to the next chapter, which is photosynthesis in higher plants. In photosynthesis in higher plants, you have obviously all the concepts about photosynthesis. Dr. Soni has answered very fast. Rohit is asking, what is your small brother's name? Are you asking about my son? My son's name is Samarth. He's almost 6 years old. His name is Samarth. All right. So, yeah. So this is the syllabus of photosynthesis. You learn about the different experiments from which he's playing outside, Sagar. I think you can hear him screaming sometimes in between, but he's playing outside. 33 to 24, Bhairavi has gone. Nice. Okay. In photosynthesis, you can expect around 2 to 3 questions every year. Deepika, I can understand Tamil. I know a lot of Tamil songs. I had a lot of Tamil friends in school, but I cannot speak very fluently. I can understand. I can manage. If I go to Tamil Nadu, I can manage, but I can't speak very fluently. Are you Tamilian? Okay, so are you ready to solve the questions from photosynthesis in flowering plants? What did I just say? Photosynthesis in higher plants. Yes, Bhairavi. Ayush, I can say Khorbo Lorbo Jeetpore, that's all. So here's our first question from this chapter. One scientist cultured cladophora in a suspension of azotobacter and illuminated the culture by splitting it through a prism. He observed that bacteria accumulated mainly in the region of violet and green light, indigo and green light, orange and yellow light, blue and red light. Blue and red light is the correct answer. Everyone's answered correctly. Naga, Nishika, Mona in the top 3. This is what the experiment looked like. Azotobacter is an aerobic bacterium. I can understand and speak decent Telugu. The one scientist that performed this experiment is our T.W. Engelmann. What he basically did was he split the light into different wavelengths and he illuminated that on cladophora, different wavelengths. And then in the same medium, he grew aerobic bacteria also. Aerobic bacteria, as you all know, will require oxygen for survival. So for respiration, oxygen is required. Wherever oxygen is released more, in those regions it will grow more. Wherever it is growing more, there more oxygen is being released. That's what it means. So wherever it is growing more, there more oxygen is released. That means in those regions more photosynthesis is taking place. This is like a very primitive type of action spectrum. But from this we got to know that the chlorophyll, the major pigment involved in photosynthesis, absorbs mostly red and blue wavelengths of light. So that is the experiment, cladophora and aerobic bacteria, right? So here is our next question. Oxygen is not produced during photosynthesis by. My mother tongue is Kannada, Sagar. Sagar is right. He's saying hydrogen sulfide. Correct. Deepika, my name is Shalini. Telugu telusandi. No, Diya, I don't know Bengali. Aditya is back. Welcome back. Okay, Naga. Towards the end of this class, I will sing one Tamil song. You've asked for it. So you should bear that torture. Oxygen is not produced during photosynthesis by green sulfur bacteria, nostoc, 𝐶𝐍𝐾𝐍, Cara. You know, plants, when we say plants perform photosynthesis, we say that they take in carbon dioxide and they give out oxygen. But the thing is, plants do not release the oxygen that they take in from carbon dioxide. Whatever oxygen is present in water, that is what will get released. Okay, but there are a couple of bacteria, like the green and the purple sulfur bacteria that perform anoxygenic photosynthesis. Over there, water is not utilized. Instead of water, hydrogen sulfide is used. In such cases, oxygen is not released. We call that type of photosynthesis as anoxygenic photosynthesis. So the correct answer here is option A. 79% of you all have answered correctly. Mona, Naga and Sagar in the top 3. So this was discovered by Cornelius van Niel. So what basically happens is, whenever the hydrogen donor is water, then oxygen will get released. Whenever the hydrogen donor is not water, it is hydrogen sulfide, then sulfur will be released, oxygen is not released. That is basically this experiment. Let's take a look at our next question from the last year's NEET paper 2023. The reaction center in PS2 has an absorption maxima at. What is absorption maxima? You kind of have like an instrument called the spectrophotometer. What we do is, we take different pigments, we isolate different pigments, and then through that you pass different wavelengths of light. So first, let's say I pass 400 nanometers of light through that spectrophotometer. Some amount of it will be absorbed, some amount of it will be released back, right? So that instrument kind of measures how much is absorbed. So in whichever wavelength maximum light is absorbed, that is your absorption maxima. So it will be different for each pigment. So here they are asking, in the reaction center in PS2 specifically, what is the absorption maxima? In the reaction center of PS2 or PS1, you always have the same pigment, which is chlorophyll A. So in PS2, the chlorophyll A has an absorption maxima at 680 nanometers. That means whenever 680 nanometer light is passed through chlorophyll A, it absorbs maximum amount of that wavelength of light. Okay, so the correct answer here is option C. 67% of you all have answered correctly. Mona, Simran and Sagar at the top 3. All of your names are starting with S. Very nice. Here is our next question from 2018. Which of the following is not a product of light reaction of photosynthesis? What happens in photosynthesis? You may study it as one equation, but it's actually two entirely different stages it happens in, just like how you prepare pani puri or golgappa. First you have like the puri made, and then you have the potato thing, and then you have the pani made, and then ultimately you will put it all together and make the pani puri. So very similarly, even here what happens is, first light energy is utilized by the pigments, water is utilized and the water splits, and ATP is produced, NADPH is produced. So in the light reaction, that is what happens. In the dark reaction, carbon dioxide fixation happens by making use of the products of light reaction, right? So which of the following is not a product of light reaction? Let's see. In the light reaction, what happens? In the light reaction, obviously sunlight is made use of, light is used, pigments are used, chlorophyll is used, and water splitting, hydrolysis of water happens. All of this is used, and then you produce ATP and NADPH in the light reaction. In the dark or the light independent phase, in the dark reaction, what happens? In the dark reaction, carbon dioxide is used, and it's used in the synthesis of glucose. In this process, okay, oxygen is also released in light reaction. These two will be used. Okay, so this is what happens during dark reaction. Which of the following is not a product of light reaction or photosynthesis? ATP is produced, oxygen is released, NADPH is produced, NADH is not. This comes in cellular respiration only. Okay, not in photosynthesis. So the correct answer here is option B. 42% have answered correctly. Sagar, Aditya, Suresh in the top 3. Next question from 2023. Right, Rohit. कब से क्लास हो रहा है? Anjali, we started at 10 o'clock. Which of the following combinations is required for chemiosmosis? You know, chemiosmosis is the way in which ATP gets produced in the light reaction. You require a proton pump, electron gradient in ATP synthase. We do not require an electron gradient, we require a proton gradient. You require a membrane, a proton pump, a proton gradient, and ATP synthase. The correct answer here is option C. So option C is the correct answer. 82% have answered correctly. Mona, Naga, and Bharat in the top 3. So this is basically what happens during chemiosmosis. Whenever electrons are being transported from the photosystem through the cytochrome B6 complex, protons are pumped from the stroma side into the thylakoid lumen. So protons will start accumulating here. Also, when water splits, hydrolysis of water happens. It happens towards the thylakoid lumen. So there is more more protons accumulating over there as well. And when during NADPH formation, NADP plus picks up protons from the stroma side. So because of that, there are fewer protons present in the stroma side. This is one thylakoid. In the thylakoid lumen, you have more protons compared to the proton concentration in the stroma. So this is the proton gradient. In order to achieve equilibrium, these protons will move out through the transmembrane portion of your ATP synthase through the F0 particle, and it comes out. As it comes out through the thylakoid lumen into the stroma, the ATP synthase head will synthesize ATP molecules. That is your chemiosmotic hypothesis. Okay. So here you require a membrane, which is this membrane. You require a proton pump. This is your proton pump. There should be a gradient of protons, and then you require ATP synthase as well, right? Now let's take a look at our next question. This is from 2022. Anjali, you have to register in Unity, you have to enroll in Unity to participate in the poll. At the top of the chat box, there is a pinned comment. There you have a link called Unity. Click on that link and enroll. It will not take more than 30 seconds. After that, you can start typing your correct option in the chat box, and it will reflect in the poll here. Okay. Which one of the following is not true regarding the release of energy during ATP synthesis through chemiosmosis? Which is not true? It involves breakdown of proton gradient, breakdown of electron gradient, movement of protons across the membrane to the stroma, reduction of NADP to NADPH on the stroma side of the membrane. Option B is the correct answer. Like we already discussed, it is not the electron gradient but the proton gradient that breaks. 75% have answered correctly, Naga, Mona and Navya in the top 3. Next question from 2020. In light reaction, plastoquinone facilitates the transfer of electrons from. The mention of plastoquinone is not there in your NCERT textbook, so this is kind of out of syllabus. But plastoquinone in photosynthesis is very similar to your ubiquinone in respiration. So what happens basically is, this is a more advanced diagram which is not given to you in your textbooks. When photosystem 2, light is incident, chlorophyll A in the reaction center, electron excitation happens, electron carrier primary acceptor picks it up, it gets excited. First electron is accepted by pheophytin, then it's passed on to plastoquinone. From plastoquinone it goes to cytochrome B6F complex. So this plastoquinone basically is transferring the electron from PS2 to cytochrome B6F complex, right? So correct answer here is option A. 92% have answered correctly, Naga, Vaishnavi and Navya in the top 3. Next question from NEET 2016. They are sister. Who is sister? In a chloroplast, the highest number of protons are found in. In a chloroplast, the highest number of protons are found in the intermembrane space, the antenna complex, in the stroma, in the lumen of thylakoids. We just now discussed, I told you how in the thylakoids the number of protons or the proton concentration is much higher compared to the stroma side. So the correct answer here is option D. Everyone's answered correctly, Naga, Nazaret and Sagar in the top 3. Next question from 2023. How many ATP and NADPH are required for the synthesis of one molecule of glucose during Calvin cycle? How many ATP and NADPH are required for the synthesis of one molecule of glucose during Calvin cycle? Everybody knows that in Calvin cycle, in the steps of reduction and regeneration, ATP and NADPH are used. For every cycle of Calvin cycle, in the reduction step, you require 2 ATPs and 2 NADPH, and in regeneration, you require 1 ATP. So totally per cycle, you require 3 ATPs and 1 NADPH, right? So per cycle, you require 2 NADPH and 3 ATP. 2 NADPH and 3 ATP. In order to form one molecule of glucose, this cycle has to happen 6 times. So multiply this with 6. For the formation of one glucose molecule, you will require 12 NADPH and you will require 18 ATP. So the correct answer to this question would be 12 NADPH and 18 ATP, which is option D. 82% have answered correctly, Naga, Mona and Sharat in the top 3. Which of the following statements is incorrect? Cyclic photophosphorylation involves both PS1 and PS2. No, it involves only PS1, not PS2. Both in ATP and NADPH plus H plus are synthesized during non-cyclic photophosphorylation. Yes, it is. Stroma lamellae have PS1 only and lack NADP reductase. Okay. Grana lamellae have both PS1 and PS2. That is correct. So the correct answer to this question is the incorrect statement, which is option A. 86% of you all have answered correctly. Jyoti, Naga and Sagar in the top 3. Next question. Okay, so this is basically cyclic and non-cyclic. For your reference, I've added it in a slide. If you're solving these questions or revising later, then you can quickly go through it. This is another question from 2015. In photosynthesis, the light-independent reactions takes place at I have not come to Hyderabad, Narayana. Not yet. In photosynthesis, the light-independent reactions take place in the stroma. Correct answer is option B. 81% have answered correctly. Vaishnavi, Naga and Sai Mona in the top 3. Next, let's take a look at this question from 2022. Yes, dark reaction happens in the stroma. Light reaction happens in the membranes of the thylakoid. Given below are two statements. Statement 1, the primary carbon dioxide acceptor in C4 plants is phosphoenol pyruvate and it is found in the mesophyll cells. Statement 2, mesophyll cells of C4 plants lack Rubisco enzyme. Okay, so which one is correct? Yes, as all of you are answering, option A is correct. In C4 plants, primary carbon dioxide acceptor is phosphoenol pyruvate. Who can tell me which is the first stable product formed in C4 pathway? Why do we call it C4 pathway? Because it is a C4 acid is formed, right? Which is that one? Who is going to tell me? Option A is the correct answer here. Naga, Navya, what? Velua. Top 3. Yeah, this is a C4 pathway. Again, for your reference in the notes, I've added this. Here's another question from 2022. Oxaloacetic acid is the correct answer. What is the role of large bundle sheath cells found around the vascular bundles in C4 plants? Generally, bundle sheath cells will not have chloroplast, but in C4 plants, even the bundle sheath cells will have chloroplast, and they will be agranule chloroplast. What is the role of large bundle sheath cells? To provide the site for photorespiratory pathway. In C4 plants, photorespiration itself will not happen. To increase the number of chloroplast for the operation of Calvin cycle, yes. To enable the plant to tolerate high temperature, that is no. To protect the vascular tissue from light. Okay, so correct answer here is option B. 79% of you all have answered correctly. Naga, Sagar and Navya, top 3. Next question. The first stable product of carbon dioxide fixation in sorghum is? Sorghum is an example for which pathway? The first stable product. Right, Sagar. The first stable product, they are not asking for which is the first, primary acceptor. There is a difference. Acceptor and product, there is a difference. This is an example for a C4 plant. So the first stable product produced there will be a C4 acid, which is oxaloacetic acid. Correct answer here is option C. 79% of you all have answered correctly. Naga, Sagar and Ansh in the top 3. Ansh, where were you all this while? Why were you not answering? Phosphoenol pyruvate is the primary carbon dioxide acceptor in C3, plant C4, plant C3 and C4. What is it? It is in C4 plants, right? So the correct answer here is option A. Naga, Sagar and Mona, top 3. Okay, here is our next question. A plant in your garden avoids photorespiratory losses, has improved water use efficiency, shows high rates of photosynthesis at high temperatures, and has improved efficiency of nitrogen utilization. In which of the following physiological groups would you assign this plant? If a plant does not show photorespiration, then it is obviously a C4 plant. So the correct answer here is option D. 72% of you all have answered correctly. Naga, Navya and Aditya in the top 3. You do understand what photorespiration is, right? Unknown girl ka chapter bhi unknown hai. So this is from photosynthesis in higher plants. Khushi is back. Khushi, khushi, khushi. Okay. Here is our next question from 2013, which is also related to C4 pathway. Bundle sheath cells are rich in PEP carboxylase, lack rubisco and PEP carboxylase are rich in rubisco. Read carefully. Bundle sheath cells. Oh, they should have mentioned that it is C4, no? Otherwise, generally bundle sheath cells, is it C3, is it C4? We don't know that. In bundle sheath cells, PEP carboxylase will not be present. It's present in mesophyll cells, right? In mesophyll, fixation, first primary fixation will happen, carboxylation will happen, then that C4 acid is transported into bundle sheath. In bundle sheath of C4 plants, they are rich in rubisco. So assuming this is related to C4 plants, we can say that it is rich in rubisco because it is in the bundle sheath cells that Calvin cycle will happen. Okay. So correct answer here is option D according to me. If it is C4, 18% have answered correctly, 19%. Mona, Sagar and Rohit in the top 3. Okay, here is another question which has occupied pretty much the entire slide. Telidu Narayana. Probably for about, we have 2 more chapters left. So until then, maybe around 40 minutes more, 30 minutes more. Okay, so what's happening here? Is this diagram clear to all of you? A is here. A is the first primary fixation that's happening. So A is fixation. Then B is here. B decarboxylation is happening. And then after decarboxylation, you have a C3 acid that's transported back and then it regenerates into PEP. So that's regeneration, fixation, decarboxylation, regeneration. Option C is the correct answer, right? Everybody knows this, right? 67% of you all have answered correctly. Simran, Naga and Sankhpal in the top 3. Correct answer is option C. Human physiology, Nishika, will happen, I don't know exactly when it will happen, but even in zoology you have it in 2 parts, 11th grade syllabus on one day, 12th grade syllabus on one day. You can check in the community post, in the community section. If the timetable is ready, it will be updated. Otherwise, you can check the live tab. You will get notifications. I mean, the live will be scheduled, I think, one day before. Like today's live, it happened at 10 o'clock, right? Yesterday only it was scheduled. So that way you can find out. Unknown girl, D is not the right answer. C is the right answer. Next question. The oxygenation activity of Rubisco enzyme in photorespiration leads to the formation of. This was in 2020. Which one, Sagar? Zoology class. One of those classes Vindhya ma'am will take, the other class Chavi ma'am will take. So during photorespiration, what happens is, this guy Rubisco is ribulose bisphosphate carboxylase oxygenase. The substrate is RUBP, a 5-carbon compound. It binds with RUBP, and to RUBP, it either adds oxygen or it adds carbon dioxide. Whether it will choose to add carbon dioxide to RUBP or oxygen to RUBP depends on their relative concentrations around this enzyme. If there's more concentration of carbon dioxide compared to oxygen, it will choose to add carbon dioxide to RUBP. RUBP is a 5-carbon compound. If you add a carbon dioxide to it, it will have 6 carbons, right? So this 6-carbon compound will split to form 2 3-carbon compounds. That is what happens during Calvin cycle. But sometimes when the carbon dioxide concentration is less and oxygen concentration is more, in such cases, the oxygen RUBP, to RUBP, Rubisco will add oxygen. In that case, you will have this 5-carbon compound splitting to form a 3-carbon compound and a 2-carbon compound. The 3-carbon compound is your phosphoglyceric acid or phosphoglycerate, and the 2-carbon compound is your phosphoglycolate, right? So in the first option, it is given 2 molecules of 3-carbon compound. It is incorrect. If you add oxygen, you don't even get 6 carbons. 1 molecule of a 3-carbon compound, 1 molecule of a 6-carbon compound we are not getting. 1 molecule of a 4 and 2 we are not getting. So 1 molecule of a 3-carbon compound is the most appropriate answer to this question. Of course, it's not only 1 molecule of 3-carbon compound. Along with that, 1 2-carbon compound also it will produce. So correct answer here is option B. 79% of you all have answered correctly. Sankhpal, Rohit and Naga have answered correctly. Next question. The process. Okay, Narayana, we'll discuss that. The process which makes major difference between C3 and C4 plants is glycolysis, Calvin cycle, photorespiration, respiration. The process which makes major difference between C3 and C4 plants is photorespiration. Whatever I told you about now, whenever Rubisco adds oxygen to RUBP instead of carbon dioxide, phosphoglycolate is formed, and it is kind of toxic. In order to eliminate that from the system, you will have to make use of ATP, and whatever carbon is fixed, that will also be used up. So it's a very wasteful process that happens in C3 plants. C4 plants have evolved and adapted in such a way that they have to avoid these photorespiratory losses. In C4 plants, because of their Kranz anatomy, because of their adaptation, anatomical adaptation, physiological adaptation, you do not see photorespiration happening in C4 plants. So that's the difference between C3 and C4. C4 plants are more productive compared to C3 plants. Correct answer here is option C. 70% of you all have answered correctly. Naga, Sagar and Sai in the top 3. Next question. A process that makes important difference between, okay, it's the same question again. Yeah, Calvin cycle will occur in both C3 and C4. In C3 plants, it happens in the mesophyll. In C4 plants, it happens in your bundle sheath cells. Yeah, C3 will happen in both. %, Naga, Simran, Sagar in the top 3. Next question from 2017. With reference to factors affecting the rate of photosynthesis, which of the following statements is not correct? It's a statement based questions, you have to find out. Yes, Rohit, correct. Which of the following statements is not correct? Increasing the atmospheric carbon dioxide concentration up to 0.05% can enhance carbon dioxide fixation rate. Yes, that is true. C3 plants respond to higher temperature with enhanced photosynthesis, while C4 plants have much lower temperature optimum. No, this is actually ultified. C4 plants have a much higher temperature optimum compared to C3 plants. Tomato is a greenhouse crop which can be grown in carbon dioxide enriched atmosphere for high yield. That is also true. Light saturation for carbon dioxide fixation occurs at 10% of full light. That is also true. So correct answer here is option B. 76% have answered Naga, Jyoti, Jyotirmay and Vaishnavi in the top 3 spots. Next question from a really long time ago when you were probably not even born, but it is still relevant, so I took it. 1996. The law of limiting factors was proposed by Liebig, Hatch and Slack, Blackman, Arnon. That's why it's called Blackman's law of limiting factors. What does Blackman's law of limiting factors say? 95% have answered correctly. Velo and Simran and Sagar in the top 3. Whenever there's any process that is controlled by more than one factor, whichever factor is near its least minimal value, that is what will influence the rate. As and when you increase the rate of that, the quantity of that is increased, the overall rate of the entire process also is going to increase. That is what is Blackman's law of limiting factors, right? So in basically you can take the example of a pen like this. In a pen, you have multiple different parts. You have this cap, you have this outer body, you have a refill inside, you have the top thing, and then you have this bottom thing. All of this is there. But when a pen is being manufactured, all of these will not be produced at once. Different components, different parts will be produced independently, and then all of them will be assembled together before it is sold in the market, right? So let's assume 100 of these caps are being produced per second, 100 of these tops are being produced per second. Everything is produced 100 per second except for the refill. The refill is produced only 10 per second. But if you have to see how many pens are being produced per second, though the rest of the components are 100 per second, it depends on the lowest one, which is the refill. So totally we'll get only 10 complete pens per second. But if you increase the rate at which the refills are being produced from 10 to 15, that will overall increase the production the rate at which this pen is overall being produced. Okay, so that is exactly how this also works. Multiple factors are, kind of influencing the rate of photosynthesis. For photosynthesis, you require light, you require carbon dioxide, you require water, you require a lot of different things. Let's say one or two of them or all of them are optimum except for one. By changing the quantity of just that one, by increasing the quantity, you can overall increase the rate of the process. Okay. Thank you, Nishika, if you found it helpful. Okay. Next question. Oh, we're done with photosynthesis. Let's take a quick look at the leaderboard. Suresh is on top of the list right now. And we have Rohit, who is second. Then we have Ram, third. Then we have Mona, Anjali, Tejaswini, Jyoti, Aditya, Soni, Sweetie, Sadhana, Naga, Vishal, Anshul Singh, Lisa, PMS, Only Studies, Tanmay, Nishika, Bharat, Sagar, Ansh, Pushpa, Mayuri. Okay, so everyone else. We have the penultimate chapter, last but one, respiration in plants. In this chapter, no concepts are deleted. Where is it 42 degrees Celsius? How are you people even surviving? I'm dying at 30 degrees only. 30, I think it's 35 today here in Bangalore, I'm not sure. Hey, if you have to talk among yourselves, no, just leave. Yeah, I said this is the penultimate. After this, there is one ultimate also. So after this chapter, we'll do plant growth and development. You are adapted. Okay. So let's start off with the questions from respiration. Is everyone ready? From this chapter, you can expect like 1 or 2 questions, but this year you might get more questions because many chapters and concepts are deleted. So this is a content, concept heavy, dense chapter. Unless you've understood it properly, you will not be able to answer the questions correctly. Here is your first question. My next class, I'm not really sure. In this series, I don't have another class, Uma Devi. Here is an assertion and reason type of question. ATP is used at 2 steps in glycolysis. Yes, it is used in 2 steps. The first ATP is used in converting glucose to glucose 6-phosphate. Yes, correct. And the second ATP is used in the conversion of fructose 6-phosphate to fructose 1,6 bisphosphate. Okay, there's still okay. So the correct answer here is option C. Both A and R are true, and R is the correct explanation of A. 42% have answered correctly. Sagar, Mona and Abhishek. Abhishek, where were you all this while? Why were you not answering? This is glycolysis. In the first step and in the third step, ATP gets used up. Glucose to glucose 6-phosphate, ATP breaks down. It gets hydrolyzed and the additional phosphate is added to the 6th carbon of glucose in the first step. In the third step, to fructose 6-phosphate, to the first carbon of fructose 6-phosphate, and other phosphate groups get added. So in the first and the third steps, ATP will get utilized. So the correct answer in the previous question was option C. Let's take a look at our next question here. In ATP, the net gain is 2 ATPs. I'm sorry, in glycolysis, the net gain is 2 ATP. The part 2 of botany session will be on the 24th of this month. All the 12th grade chapters will be covered on that class, in that class. Conversion of glucose to glucose 6-phosphate. The first irreversible reaction of glycolysis is catalyzed by which enzyme? By hexokinase. It is a memory based question. Nothing to explain here. Correct answer is option C. 60% of you all have answered correctly. Sangpal, Sagar and Anvay are in the top 3. Next question, 2018. What is the role of NAD in cellular respiration? Helicopter kinase. Which one is the energy yielding process? Among which, what are your options? Which one means respiration itself will yield energy? What is the role of NAD in cellular respiration? It functions as an electron carrier, right? So whenever oxidation, redox reactions are happening, it will accept the electron and pick up the protons as well and get reduced to form NADH, which is later used in ATP synthesis. Correct answer here is option B. 57% of you all have answered correctly. Samiraj, Mona, Matti Khordor are in the top 3. Next question from 2022. What amount of energy is released from glucose during lactic acid fermentation? This again is a direct statement from your NCERT textbooks. In fermentation, be it lactic acid fermentation or alcoholic fermentation, glucose is not fully broken down. It's only partially oxidized. So when partial oxidation happens, all of the energy in glucose will not be released. In lactic acid fermentation specifically, less than 7% of energy is released, and even that less than 7% is not entirely trapped in the form of ATP. So very little energy is, very few molecules of ATP can be obtained from lactic acid fermentation. So the correct answer here is option D. 68% have answered correctly. Sanya, Mona and Aditya in the top 3. Next question from 2014. In which one of the following processes carbon dioxide is not released? Regardless of whether aerobic respiration happens in plants or animals, carbon dioxide for sure will be released in aerobic respiration. Alcoholic fermentation, what happens? You have a C3 pyruvic acid that gets converted to ethyl alcohol that has 2 carbons, and the other molecule of carbon dioxide will be released. In lactic acid fermentation, C3 pyruvic acid gets converted into a C3 lactic acid. Here there is no carbon dioxide release. So correct answer here is option D. 68% of you all have answered correctly. Naga, Raju and Rohit in the top 3. Here I've added the equations of alcoholic fermentation and lactic acid fermentation for your reference. Both in alcohol and lactic acid fermentation, no? What typos? Be more careful, Aditya. Here is another question from last year's NEET 2023. Match list 1 with list 2. Here they are asking about the enzymes required. Spurti is in Lakshya KCET batch. You sent post, it cannot be post and stay. Okay, did you contact the help desk? Spurti? Is there any issue that you're facing? Oxidative decarboxylation, where does it happen? Oxidative decarboxylation is basically your link reaction. Glycolysis is also called EMP pathway. Oxidative phosphorylation happens in electron transport system. Pyruvate dehydrogenase, tricarboxylic acid cycle requires citrate synthase. So correct answer is A2, B4, C3, D1, which is option B. Any doubts in this question? It's pretty straightforward. Only 2 people have answered correctly, Bharat and Rohit, so obviously there is doubt. Oxidative decarboxylation and oxidative phosphorylation are two entirely different things. Oxidative decarboxylation is your link reaction which will require the enzyme pyruvate dehydrogenase. Oxidative phosphorylation is the production of ATP using the electron transport chain in your mitochondria, electron transport system in your mitochondria. So that is electron transport system. Glycolysis is also called EMP pathway, named after the scientists, after the researchers who elucidated that pathway, Emden, Meyerhoff and Parnas. Then we have tricarboxylic acid cycle, tricarboxylic acid cycle, TCA cycle, or your citric acid cycle, or your Krebs cycle is the same thing. Citrate synthase is the enzyme that is required for the formation of citric acid from the condensation of oxaloacetic acid and your acetyl-CoA. So that is the correct match. The correct answer here is option B. All right. Next question. Spurti, what exactly do you need? The number of substrate level phosphorylations in one turn of citric acid cycle is 0, 1, 2, 3. One turn of citric acid cycle, how many? One turn. Do you understand what substrate level phosphorylation is? Phosphorylation is basically addition of a phosphate group to a substrate. We talk about phosphorylation in terms of formation of ATP. There are multiple ways in which ATP can be formed. Photophosphorylation you've learnt in photosynthesis. In the light reaction, sunlight energy is used and that energy is used to trap, that energy is used in the synthesis of ATP. That is photophosphorylation. Substrate level phosphorylation we've seen in glycolysis. What happens? From one substrate to another, phosphate groups will get transferred and ATP synthesis happens. So that is substrate level phosphorylation. Oxidative phosphorylation happens in the inner mitochondrial membrane in your electron transport system. That is your oxidative phosphorylation. Here the question they've asked is, in your Krebs cycle, in one cycle of, Krebs cycle, in one turn of your Krebs cycle or your citric acid cycle, how many substrate level phosphorylations will happen? Only one. Right? So 67% of you all have answered correctly. Naga, Abhishek and Bharat are in the top 3. It is in this step that substrate level phosphorylation will happen. When succinyl coenzyme A is getting converted to succinic acid, a GDP molecule gets converted to GTP. After that, it's immediately coupled with ATP synthesis. So it is in this step that substrate level phosphorylation will happen. Om Prakash, I cannot speak very fluent Hindi unfortunately, and there are many students in this class who follow this channel who cannot understand Hindi. This channel itself is for people who can't understand Hindi and who would prefer to learn in pure English. So I'm sorry, I cannot help you with that right now. I'm sure there are a lot of other PW channels where they teach in Hindi. I hope you can find one such channel. Here is our next question from 2019. There is one PW Gujarati channel also, in case you don't already know. Where is respiratory electron transport system located in plants? Is it in the mitochondrial matrix? Is it in the outer mitochondrial membrane, inner mitochondrial membrane, intermembrane space? Where is it located? It's in the inner mitochondrial membrane. It's for the exact same reason that you see so many infoldings of your inner mitochondrial membrane. Because the infoldings increase the surface area available, you can accommodate more of those complexes, more of those ATP synthase enzymes, so that maximum amount of ATP is synthesized, right? So the correct answer to this question here is option C. 74% have answered correctly. Ching and Mona and Anvay are in the top 3. Next question from 2018. For which question are you talking about, Aditya? The previous one, can you mention specifically which question you're talking, which question you're referring to? Which of these statements is incorrect? Enzymes of TCA cycle are present in the mitochondrial matrix, yes. Glycolysis occurs in the cytosol or the cytoplasm, yes. Glycolysis operates for as long as it is supplied with NAD that can pick up hydrogen atoms, yes. Oxidative phosphorylation takes place in the inner mitochondrial membrane, this is incorrect. So correct answer here is option D. Not many people have answered this correctly. Praveena, Jyotirmay and Sanya are in the top 3. Aditya, which question are you talking about? ETS? Let me check. No, how can it be D? It's not present in the space, right? It's all those complexes are present in the inner mitochondrial membrane. How can it be D? Okay, let's move on and take a look at our next question related to Krebs cycle. This is from 2017. Which statement is wrong for Krebs cycle? There is one point in the cycle where FAD is reduced to FADH2. This is correct. During the conversion of succinyl coenzyme A to succinic acid, a molecule of GTP is synthesized. Yes. The cycle starts with the condensation of acetyl group, which is acetyl-CoA, with pyruvic acid to yield citric acid. No, this is not pyruvic acid. This is oxaloacetic acid. Acetyl coenzyme A has 2 carbon, oxaloacetic acid has 4 carbon, it will form a citric acid which has 6 carbons. So correct answer here is option C. This is wrong. There are 3 points in the cycle where NAD plus is reduced to NADH and H plus. This is correct. Okay, so incorrect one here is option C. Very few of you have answered this correctly, just 27%. Vaishnavi, Anjali and Sanghpal are in the top 3. What is the confusion here? Which option did you all think it is correct? Yeah, Sanya. Gautam, you have to, there is a link given at the top of the chat box. It's called Unity link. It's a portal where you will have to enroll. You just have to click on that link and enter a few credentials. After that, you can just start typing your correct, whichever option you think is correct, just start typing it in the chat box. Then it will pick it up. It will reflect in the leaderboard. Yeah, moe only. Why not B? Deepika is asking for this question. GTP synthesized. No, it is true. No, we saw here. Succinyl-CoA to succinic acid, GTP synthesized. They've asked us to find out which is incorrect, not which is correct. Right? So, the incorrect statement here is this. Next question from NEET 2016. Oxidative phosphorylation is formation of ATP by transfer of phosphate group from substrate to ADP. That would be substrate level phosphorylation, not oxidative phosphorylation. This is incorrect. Oxidation of phosphate group in ATP, no. Addition of phosphate group to ATP, no. Formation of ATP by energy released from electrons removed during substrate oxidation, yes. This is what oxidative phosphorylation is. Correct answer is option D. 33% of you all have answered correctly. Sai, Sagar and Deepika in the top 3 spots. Yeah, so this is, I've added this here just so if you're referring to this notes, you get the information from this. Okay. Next question. In mitochondria, where do the protons accumulate? Outer membrane, inner membrane, intermembrane space, matrix. It accumulates in the intermembrane space right here. As and when electrons are being transported from one electron carrier to the other, from one complex to the other, protons will get pumped from the matrix side to the intermembrane space side. So in the intermembrane space, protons will start accumulating. So highest concentration of protons in the mitochondria will be in your intermembrane space. If you think about chloroplast, more proton concentration will be in your thylakoid lumen. In mitochondria, it will be in your intermembrane space. Alright. So the correct answer here is option C. And afterwards, it is in the matrix that you will release the, into the matrix that the ATP will get formed. 37% of you all have answered. Sagar, Naga and Mona are in the top 3. Mona is doing a great job today. Here is our next question from the year 2023, very latest question, brand new. In which one of the following do the two names refer to one and the same thing? Krebs cycle and Calvin cycle are not the same thing. Not at all. No. Tricarboxylic acid and citric acid, both of these are the same. Tricarboxylic acid cycle, citric acid cycle, TCA cycle in short, and also Krebs cycle, all of these are the same thing. Okay, it's called tricarboxylic acid cycle because citric acid, which is the first stable compound, a 6-carbon compound, has 3 carboxylic acid groups. So that is why it's called a tricarboxylic acid cycle. Alright, so correct answer here is option B. Citric and Calvin are not the same thing. Tricarboxylic acid and urea cycle are not the same thing. So correct answer here is option B. 47% of you all have answered. Sai, Nishita, Sai and Deepika in the top 3. Next question. What is the net gain of ATP when each molecule of glucose is converted to 2 molecules of pyruvic acid? They haven't asked directly what is the net gain of ATP in glycolysis. They've mentioned what happens in glycolysis. You know that in glycolysis there is incomplete oxidation of glucose. In 10 steps, glucose gets broken down into 2 molecules of pyruvic acid. So what is the net gain of ATP there? In the first 5 steps, 2 ATPs are utilized. In the next 5 steps, 4 ATPs are synthesized. So the net gain would be 2 ATP. Correct answer here is option C. 76% have answered correctly. Naga, Sanya, Nishita, Sai in the top 3 spots. This is where one ATP gets utilized, another ATP gets utilized. But here in 2 steps, 2 ATPs are formed here, 2 ATPs are formed here. So totally 4 are formed, 2 are utilized. So the correct, the net gain would be 2 ATPs. Okay, so let's take a look at our next question here, which is from NEET 2021. Naga, you think it's 3 ATP? Net gain is 2. Which of the following statements is incorrect? Oxidation reduction reactions produce proton gradient in respiration. Correct. During aerobic respiration, role of oxygen is limited to the terminal stage. Correct. In ETC, 1 molecule of NADH plus H plus gives rise to 2 ATP molecules and 1 FADH2 gives rise to 3 ATP molecules. No, they have ultified this. NADH will give 3 and FADH2 will give 2. So that is incorrect. ATP is synthesized through complex 5. Correct. The other name for it is ATP synthase itself. So this also is a correct statement. What is not correct is option C over here. So the correct answer here is option C. Naga, Jyotirmai and Nishchita Sai have answered very quickly. They are in the top 3 spots. Alright. Okay. Next question. Yeah, so basically what happens here is NADH will pass on the electron to complex 1 and from complex 1 to complex 3, from complex 3 to complex 4 and from complex 4 finally to oxygen, right? This is how the flow is. So as electrons are being passed on from first complex to in this sequence, protons will be pumped from the matrix side to the intermembrane space via the complex 1 and complex 4. Complex 3 and complex 4. 1, 3, 4. In all of them, protons will be pumped. So through the first complex, 4 protons, through the third complex, 4 protons, through the fourth complex, 2 protons will be pumped. So totally you're getting like 10 protons from one molecule of NADH. So according to your newly revised NCERT, actually this is very controversial. Different books give different numbers, but according to your newly, modified, updated NCERT, for every ATP molecule, 4 protons will have to pass through this. So from one NADH, you're getting 10 protons. If 4 protons will give rise to 1 ATP, then from 10 protons, you get approximately 2.5 ATP, which we round off to 3 ATP. But if you look at how FADH2 will pass protons, FADH2 is from the second complex to the third complex to the fourth complex and finally to oxygen, right? So over here, the second complex is not a proton pump. Through the second complex, no protons are being pumped. From the third complex, 4, from the fourth complex, 2. So totally 6 of them will be pumped. If every 4 ATPs, if every 4 protons will give rise to 1 ATP, then from FADH2, you'll get around 1.5 ATP, which we round off to 2 ATPs. Okay, so per FADH2 you get 2 ATP, per NADH you get 3 ATP. Okay, now let's move on to our next question. We're almost done with respiration in plants. We have like 2-3 more questions only. Not much longer. Maybe we'll be done by 4 o'clock, hopefully. Which of the following biomolecules is common to respiration-mediated breakdown of fats, carbohydrates, and proteins? This was asked in NEET 2016 phase 2. The correct answer here is option D, acetyl-CoA. 61% have answered correctly. Deepika, Sagar, Nishchita, Sai in the top 3 spots. So you have seen this kind of flow chart in your textbooks, right? So if at all proteins and lipids have to enter the respiratory pathway, how will they enter? Of course, they will not start as glucose, right? So they have to come in between in some other, through some other way, through some other intermediate. So the question that we had here was, which of the given options biomolecules is common to respiratory mediated breakdown of fats, carbohydrates, and proteins? So the correct answer is acetyl-CoA. Proteins can enter the pathway through acetyl-CoA, fats can enter the pathway through acetyl-CoA, and acetyl-CoA is one of the intermediates in the breakdown of carbohydrates as well. So correct answer is acetyl coenzyme A. The next question that we have here is from 2019. Narayana, 1 ATP, 4 protons according to your latest updated NCERT. Earlier they had given 2 protons, but this time they've changed it in the text, but in the diagram it is still showing 2 protons only. Respiratory quotient or RQ value of tripalmitin is what? 0.09, 0.9, 0.7, 0.07. First of all, what is respiratory quotient? We also call it the respiratory ratio. It is basically the ratio of how much carbon dioxide has evolved to how much oxygen is consumed in the process of respiration. If you take tripalmitin as example, in the process of respiration, 145 molecules of oxygen is utilized and 102 carbon dioxide molecules are released. So 102 divided by 145 is 0.7. So the correct answer to this question is option C, 0.7. For glucose, it is 1. 83% have answered correctly. Naga, Nishchita, Sai and Sanya in the top 3. Let's take a look at our next question. Wait, we are done. Let's take a look at the leaderboard. We have Suresh on top, Rohit Gupta second, Then we have Ram in the third position. Then we have Mona, Anjali, Tejaswini, Jyoti, Aditya, Soni, Naga, Sweetie, Sagar, Sadhana, Vishal, and everyone else. Yeah, sure. We'll do it in the end. Thanks, Vaishnavi, if you found it useful. Narayana, can you get 380 plus out of 720 you're asking in 15 days? Now we have plant growth and development. This is the syllabus. In plant growth and development, 3 concepts have been deleted. That is your photoperiodism, vernalization and dormancy. Everything else is there, retained as it is. In the first part, you learn about growth and different features of growth, different attributes of growth. In the second part, you learn about plant growth regulators exclusively. What are the different hormones that influence the growth and development in plants? What are its different physiological effects, and all of that is what this chapter basically deals with. In this chapter, mostly questions are asked from your hormones part. Okay, for mostly it is asked from hormone part. Okay, so from this chapter again, you can expect around 2 to 3 questions. But like I've been mentioning since the beginning of the class, since many concepts and many chapters have been deleted, it's likely that you can expect more number of questions from this chapter. Okay, so is everyone ready to solve the questions from the last chapter for today? Let me know in the comments. Give me a thumbs up if you're ready. The process of growth is maximum during which phase? Very simple question. It is in, oh my god, what have I done? In the log phase, option A. In the log phase, you can see exponential growth. Jyoti, Naga and Nishchita Sai have answered very quickly. They are in the top 3. 78% of you all have answered correctly. This is a question from 2020. So when you look at how exponential growth happens, you get a sigmoid curve basically. When you're plotting the size of an organ against time, in the sigmoid curve, you have a period where growth is happening, but it's happening at a very slow rate. Eventually, growth will happen at a very rapid rate. After that, it will kind of become stationary. So maximum growth happens in the exponential phase. It is also called the log phase. Okay, so the correct answer to this question is log phase. Here is another question. Deepika, please try to check your internet connection. Is there anybody else facing issues with audio or video? Is it Deepika only? Anyone else is facing issues? Let me know. In tissue culture experiments, leaf mesophyll cells are put in a culture medium to form callus. This phenomenon may be called as, this is the latest NEET exam 2023. Mesophyll cells, they are a type of parenchyma cells. Are parenchyma cells differentiated or undifferentiated? Parenchyma cells are differentiated cells. What is callus? Callus basically is undifferentiated mass of cells, right? So what happens is you have meristematic cells. These meristematic cells are constantly undergoing division. Meristematic cells. At one point, one subset of meristematic cells will stop undergoing cell division. It will take up specific permanent functions in the plant. It will undergo modifications accordingly and become permanent tissues. This process where meristematic tissues stop dividing and take up permanent function is called differentiation. This permanent tissue could be, it could be parenchyma, it could be collenchyma, it could be sclerenchyma, or whatever type of permanent tissue, right? This process is called differentiation. Now, just like how sometimes most of us take up certain careers, and for the rest of our life we stick with it. Like I'm a teacher right now, that is my permanent tissue. You can think I'm differentiated. Earlier I had many options. When I'm meristematic, when a tissue is meristematic, it can become more than one. I mean, it can become any one of many things. I could have become a chef, I could have become a musician, I could have become a teacher, I could have become a doctor myself, but I chose to become a teacher and I've stuck with that profession for now. So that's what happens with most people, right? One profession they choose, one career they choose, they stick with it for the rest of their lives. Majority of your permanent differentiated tissues are like that. Once they become parenchyma, they don't really change their role after that. Most of the tissues are like that. But sometimes some permanent tissues will regain the ability to divide. What is differentiating this meristem and permanent tissues? Meristematic tissue has the ability to divide, it is not differentiated. Permanent tissue has lost the ability to divide. Sometimes the permanent tissues will, some of them, sometimes in certain conditions, will regain the ability to divide and become meristematic again. This process is called de-differentiation. Like for example, today I decide that I don't want to be a teacher anymore, and from tomorrow I want to pursue something else. Like I want to be a hairdresser, let's say. So that is me de-differentiating. Okay. After de-differentiating, whatever de-differentiated meristem is formed, there that meristem can further become any permanent tissue. Okay, again permanent. Initially it probably had differentiated to become parenchyma, but right now it's changed its mind. It's gone back to becoming meristematic. Right now it wants to become something else, probably collenchyma, probably some element of xylem. Whatever it is becoming permanent again. Now it is differentiating yet another time. We call this re-differentiation. Like I had differentiated from a girl who knew nothing, who had so many options in front of her, but I chose teaching. That is me getting differentiated. I re-differentiated, came back and looked at my other options. I de-differentiated, came back and looked at my other options. And then I chose to become a hairdresser again. So that is me re-differentiating. It's something very similar. Okay. So over here, we had parenchyma, which is an already differentiated type of cell, already differentiated type of tissue. This has gone back to become something that is not differentiated, which is callus. This process is called de-differentiation. The correct answer to this question here is option D. Naga, Nishchita Sai, Mona, and yeah, you have answered very quickly in the top 3. 53% of you all have gotten this correctly. Is this clear now? There's something wrong with my digital board. I think there's some problem with the lights, maybe. It's part of the syllabus. Anything can be asked from your syllabus. I don't know what happened, really. What is the problem? What is your problem? Here is the next question. Which one of the following plants does not show plasticity? Which one of the following plants does not show plasticity? What is plasticity? Depending upon the environmental conditions, depending upon the phase in which it is in its life cycle, plants adopt different developmental pathways. That is what plasticity is. Coriander, cotton, buttercup, all of these show plasticity. Maize does not. So correct answer here is option D. Maize is not an example for plasticity. Nishita, Sanya, and another Nishita Sai have topped. I mean, they're in the top 3. So I've added a slide here based on that. This following different developmental pathways can happen due to environment or due to a different phase in its life. Example for how different development pathway happens in different phase in life. You can take the example of coriander. Everybody's house you bring coriander, right? Next time you take a look at your coriander leaves very carefully. The young leaves will be more dissected and the older leaves will be kind of more full. So you can see a distinct difference in the type of leaves towards the tip and towards the base. That is plasticity. In different phase of its life, it's having a different morphology. Then response to environment. Take the example of buttercup, which is an aquatic plant which can also grow on terrestrial habitats. So whenever it's produced in water habitat, its leaves will be highly dissected like this, and on terrestrial habitat the same species plant will produce leaves like this. So according to its environment also, its developmental pathway will vary. Okay, so that is that. Here is our next question, which is very simple. You should take like 3 seconds to answer this. Plants follow different pathways in response to environment or phases of life to form different kinds of structures. This ability is called. Plants follow different pathways. This is called plasticity. We just now discussed this. Option D is the correct answer. 75% of you all have answered correctly. Mona, Aryan and Nishita are in the top 3. Now let's start off our questions related to hormones. Which hormone promotes internode petiole elongation in deep water ice? It's very, very important for you to memorize the physiological effects of all hormones. Okay, you're Nishika. Did I call you Nishita? I'm sorry. The correct answer here is ethylene. Option A, like most of you have answered. 42% have answered correctly. Naga, Aryan and Mona in the top 3. I've added a slide here where I've listed out all of the effects of ethylene so that you can revise very quickly. Next question here. Spraying which of the following phytohormones on juvenile conifers helps in hastening the maturity period that leads to early seed production? In some countries, the seeds of conifers is consumed as a delicacy. So if its maturity period is speeded or hastened, then it will produce seeds more quickly. So you can reap benefits more quickly. For that reason, gibberellic acids are used. So the correct answer here, I will mark it here, just a minute, in a different color. So the correct answer here is option D. 82%, 83% of you all have answered correctly. Naga, Sai and Sanya are in the top 3. Next question here from 2022. The gaseous plant growth regulator is used in plants too. Which is the gaseous plant growth regulator? It is ethylene, right? What is its function? Speeding up the malting process? No. Promote the root growth and root hair formation? Yes. Help overcome apical dominance? No. Kill dicotyledonous weeds? No. So correct answer here is option B. That is one of the roles of ethylene. 78% have answered correctly. Naga, Deepika, Abhishek in the top 3. Again, I've added a slide here. You can see here this point. Next, one question from 2022. Production of cucumber has increased manifold in recent years. Application of which of the following phytohormones has resulted? Just give me a minute. It is ethylene. Option C is correct. 73% of you all have answered correctly. Naga, Aryan and Ronak. Why are you so angry, Ronak? Are in the top 3 spots. I've added that also. Ethaphon, you know, ethylene is a gaseous plant growth regulator. How do you administer that to plants? How do you use it on plants if you have to reap the benefits of the physiological effects of ethylene? You get an aqueous solution of it in the form of ethaphon. You spray this on plants, the plants will absorb it, and inside the plant, ethylene will be released in very small quantities. So, some of the uses of that is it fastens or hastens the ripening of fruits in tomatoes and apples. Abscission of flowers and fruits also, it helps. It promotes female flowers in cucumbers. This was the question. So that also, that is the correct answer. I think I've already marked it. Okay. Next. Plant hormone used to destroy weeds in a field is? What is it? IBA is indole butyric acid, IAA is indole acetic acid, naphthalene acetic acid, 2,4-D is 2,4-dichlorophenoxyacetic acid. The plant hormone that is used to destroy weeds in a field is 2,4-D. It is used as a weedicide. It is basically an auxin. Correct answer is option D. Aryan, Naga and Simona are in the top 3. So basically what this 2,4-D does is it kills specifically dicot plants. If you have a field where you're growing monocot plants and you have unwanted dicot weeds growing in that, in order to specifically target and kill dicot plants, this can be used. In fact, it was used also during wars in Vietnam. It is also called Agent Orange. Okay. What is your doubt, Deepika? I don't see what doubt it is. If you can type it again, I can answer. Name the plant growth regulator which upon spraying on sugarcane crop increases the length of the stem, thus increasing the yield of the sugarcane crop. Generally, if there's any any place where internodal elongation is happening or something is increasing in length, like the floral axis is increasing in length or bolting is happening, generally it is the gibberellic acids. Okay, so even here it is gibberellic acid or gibberellin that is used to increase the length of sugarcane. In sugarcane, what is the commercially important part? It is the stem, right? So in the stem, if the internodes are lengthier, if they are longer, then you get more sugar from it, or you can get more sugarcane from it. So commercially, it's better for you if the internodal length is more. If you go to buy sugarcane during Sankranti or Pongal, you will know. When you look at these sugarcanes to buy them for your festival, the first thing that you want to see is the notes should not be too close to each other. The notes should be farther away. That is the first thing you check for when you're buying sugarcanes for your house, right? So it is better if the internodal length is more, because you get more sugar in that case, and it's commercially important. So that can be achieved with gibberellin. So the correct answer to this question is option B. 78% of you all have answered correctly. Naga, Mona and Jyoti in the top 3 spots. I've added a slide for this. Where is it? Sugarcane. Here you are. Okay, fine. Next question from 2020. Which of the following is not an inhibitory substance governing seed dormancy? Which of the following is not an inhibitory substance governing seed dormancy? Abscisic acid also governs seed dormancy. Gibberellic acid is also governs seed dormancy. But here they are asking which is not inhibitory. Abscisic acid is inhibitory, gibberellic acid is not. So correct answer here is option A. 67% have answered correctly. Naga, Nishika and Mona are in the top 3 spots. Next, we have a question from 2019. It takes a very long time for pineapple plants to produce flowers. Which combination of hormones can be applied to artificially induce flowering in pineapple plants throughout the year to increase yield? It takes a very long time for pineapple. Actually, in your physiological role, in your textbook, there is a mention of pineapple 2 times in 2 different plant growth regulators. This question is based on that. It is auxins and ethylene. Option B is the correct answer. 60% have answered correctly. Sweetie said you were going and you said Tata bye bye long time ago. You're still there or you came back just now. Sweetie 690 and Naga and Mona are in the top 3 spots. I've added a slide here so you can mark it here. Okay, so this promotes flowering in pineapples, and in ethylene it synchronizes fruit set in pineapple. So both the 2 of them can be used together. Okay. Next question. Fruit and leaf drop at early stages can be prevented by the application of which hormone? Ethylene, auxin, gibberellic acid, cytokinin. Auxins prevent the fruit and leaf drop at early stages, but they will promote abscission after senescence. Okay, so in the early stages it prevents fruit and leaf drop. So correct answer here is option B, auxins. 88% have answered correctly. We have Aryan, Naga and Mona in the top 3 spots. Have I added a slide for auxin? I guess not. Okay, I've added one here. You can see here, it prevents early leaf and fruit drop, but it promotes abscission of mature leaves and fruits. So in the question we were asked, early stages, leaf and fruit drop, which hormone will prevent? So the correct answer is auxins. Next question here. You are given a tissue with its potential for differentiation in an artificial culture. Which of the following pairs of hormones would you add to the medium to secure shoots as well as roots? This is NEET 2016, phase 2. Indole acetic acid and gibberellin, auxin and cytokinin, auxin and abscisic acid, gibberellin and abscisic acid. Option B is the correct answer. Whenever you have to grow new plants using tissue culture, we give them a growth medium. For tissue culture, the explant that you take could be any plant part. It could be a leaf, it could be a stem, it could be the root, it could be the pollen grain, it could be anything. Okay, so if you take that explant and add it into the nutrient medium, it will first form a callus, which is an undifferentiated mass of cells. If you want that to become differentiated to produce roots and stem, you will have to add these hormones for that. You will add auxins and cytokinins. Auxins basically helps in initiation of root system. It will help in rooting, and the cytokinins will help in the development of the shoot system. So auxins and cytokinins have to be added. Option B is the correct answer. 85% of you all have answered correctly. Naga, Mayuri and Abhishek in the top 3. Before this class gets over, I wish to see at least one question where 100% of you all will answer correctly. What causes a green plant exposed to light on only one side to bend towards the source of light as it grows? Everybody may have performed this experiment or read about this experiment in school. Like you have one dabba, one closed opaque box. On one side of that box you would have cut out a hole, and then you keep a plant inside it and you keep it in the sunlight, and then the plant kind of bends towards sunlight, right? So why does that happen? That happens because at the tip you have your auxins. If the sunlight is in this direction, the auxins will accumulate in the opposite direction. This auxin will help in the growth of cells, right? It helps in increasing the length of the cell. Elongation of the cell also will happen. It will promote cell division also. So the cells present on this side will start growing, elongating much longer, much quicker than the cells on the illuminated side. So on this side it will kind of cause the stem tip to bend. Okay, so light stimulates plant cells on the lighted side to grow faster. Light does not stimulate. Auxins accumulate on the shaded side, stimulating greater cell elongation there. This is the correct answer. Green plants need light to perform photosynthesis. Yes, they do, which is why they bend, but that is not the reason why that bending happens. Green plants seek light because they are phototropic. Yes, that is true, but that's not the reason why it bends. The reason it bends is because auxins are accumulating on the shaded side, and because cell elongation happens at a much rapid rate on the shaded side, it bends towards the sunlight. Okay, so option B here is the correct answer. 71% of you all have answered correctly. We have Gokul, Sagar and Nishika in the top 3 spots. Here is our next question. Which one of the following generally acts as an antagonist to gibberellins? Zeatin is example for cytokinin, ethylene, abscisic acid, indole acetic acid. Which acts as an antagonist to gibberellins? It is abscisic acid. Option C is the correct answer. 27% of you all have answered correctly. Aryan, Gokul Chandra and Nishika are in the top 3 spots. Yeah, so here I have added a slide. Yeah, abscisic acid acts as an antagonist to gibberellic acids in most cases. So with this, we have completed all the important PYQs from your plant growth and development chapter. We've come to the end of today's session. Let's take a quick look at the leaderboard. Congratulate all the people who have consistently answered from the beginning. Okay. Suresh is on top of the list. Congratulations, Suresh. Suresh is pretty much in every class in the top 3 from the NEET 50-day challenge also. I hope you do really well in your exams. Your preparation is really great. Then we have Rohit, who also is doing pretty well. And then we have Mona, who's answered most questions correctly. Ram, I think he stopped answering somewhere in between the class and he left, which is why he was giving a very tough competition to Suresh in the beginning. Then we have Ram and then Anjali. Every one of you, congratulations. Tejaswini, Jyoti, Aditya, Naga, Soni, Nishika, Sagar, Sweetie, Sadhana, Vishal, Aanchal, Lisa, all of you who have been answering consistently, congratulations. Maybe after this session, you know what are your weak areas, what questions you got wrong, what questions you got incorrectly, why you answered it incorrectly. Find out what your weakness is, find out what your weak area is and work on it and do exceptionally well in your NEET exam. And before NEET exam, I may not get another class with you. So this might be officially my last class with you all before NEET 2024. So one more thing that I want to tell you all is keep your mindset very positive before NEET exam. Don't enter the exam hall with a state of anxiety or fear or nervousness. Think you worked so far, so so hard all this while. Take all of that knowledge that you've gained through these years into the exam hall, because sometimes students who may have not studied so well, with the right application of their mind, with the positive mindset, I have seen in my students themselves how people who have a positive mindset, who were average students, score better than students who studied a lot, who had a lot of knowledge but got anxious and scared at the time of exam and they could not perform well. I do not want you to be one of those students. So whatever you've studied, all this while, use it to the best of your ability. Go to the exam hall with a positive mindset. After the exam is over, I want to hear from you all how much you scored. So come back and drop your comments, your score, your mark, how much you're expecting, how well your exam went in the comment section in this video. So I will come back and check it and I will feel happy if you all have done well. So yeah, that's that. And thank you all for attending today's session. The notes will be available very soon in the description box as a PDF link. You can download that and use it for your revision. And yeah, all the very best. Thank you. Yes, I promised I will sing a song. I will sing a song, but because I've been talking since the last I don't know how many hours, I may sound very bad, but okay, I'll sing one Tamil song. I'll tell you a story about this Tamil song that I'm about to sing. I think I was your age, probably 11th grade or 12th grade, and then one movie had released. It was like crazy craze for that movie. It's called Vinnai Thandi Varuvaya. Okay. So there was one very popular song in that movie. Back then, I didn't have Google and mobile phones and smartphones, so I could download the lyrics and learn the song. I had a best friend in college who was a Tamil speaking person. So we heard this song on the radio. We recorded it on our tape recorder, and then I asked her to teach me that song. I wrote it down in my notebook at the back of my notebook, and I learned that song. So yeah, 3, 2, 1, begin. I don't know, I may pronounce the words very incorrectly because I don't speak Tamil. அந்த நேரம் கண் பார்த்து கந்தலாகி போன நேரம் ஏதோ ஆச்சு உவான தீண்டி வந்தாச்சு அப்பாவும் திட்டி எல்லாம் காற்றோடு போய் போச்சு ஹொசானா என் வாசல் தாண்டி போனாலே ஹொசானா வேறொன்றும் செய்யாமலே நானாகி போகிறேன் சுற்று நூறாகிறேன் அவள் போன பெண் பயந்தை நெஞ்சில் தேடி போகிறேன் ஹலோ ஹலோ. That's it. It's called Hosanna. It's from a movie called Vinnai Thandi Varuvaya. Thank you very much for attending my musical concert. Bye bye. All the best.